everyone! Would you believe me if I told you that on the same planet that we live on, there are also organisms that kind of resemble small glass pieces? Also, there are organisms that kind of glow in the dark. There are some of them which kind of suffer from an identity crisis and can't decide whether they want to be more like plants or like animals. And in this chapter, Biological Classification, we are going to deep dive into the world of living organisms, find out about their different features, and try and classify them into distinct groups. Through the course of this chapter, we're going to come across a lot of different types of organisms. We'll have to memorize the characteristics and examples. And I'm going to make your preparation smoother by sharing the class notes along with TPP questions to practice on the PW app, where there is a free batch called Complete Botany by Shalini Ma'am. You may access the batch using the link given in the description below. And while you're on the app, make sure you check out all of the amazing features there, which is going to help you reach your goal of becoming a doctor. Now, without any further delay, let's get the session started. First things first, should you refer the old NCERT or the new one for this chapter? I would suggest that you refer the latest edition of NCERT for this one, because in some of the older books, there are some scientist names mentioned under the virus topic which have been mixed up. So it's best you stick to the new edition of NCERT. The name of the chapter itself is Biological Classification. So it's obvious that we're going to learn about how living organisms are classified. So very early on, people did attempt classifying living organisms, but back in the day, since people did not have that much of a scientific temperament, there was not much of practicality or scientific basis for classifying living organisms. It was mostly not scientific. And as NCERT says, it was done instinctively. Instinctively means without putting conscious thought into it. So back in the day, very early on, humans classified living organisms not based on its morphology or anatomy like we do today, but it was mostly based on how useful that particular organism was for humans. For example, if you consider like a plant or animal, they thought about whether that plant could be used as food, whether the fiber that from that plant could be used as, clothing, or the fur from the animal could be used as clothing, whether that animal could be consumed. If they come across a tree, they would think about whether the wood from that tree could be used to build houses. So mostly classifying living organisms was based on how it was useful for humans, either for food, for shelter, or for clothing. As time passed, humans became more and more intelligent, and certain amount of scientific temperament development happened in humans. So then they started classifying living organisms using some scientific criteria. One of the very first people to do that was Aristotle. Everybody has heard about Aristotle, right? He was a very famous Greek philosopher. He used some scientific basis for classifying both plants as well as animals. One example for his classification was using very simple morphological features to classify plants. He classified plants into three groups, which is herbs, shrubs, and trees. And you all must be like, does this even count as a scientific classification? This is something that a second grade kid would know. So yeah, obviously for today's time, this may not seem very significant, but back in the day when people did not even have a scientific basis for classification, he at least used some simple morphological characters for classifying. So basically, he looked at whether the stem was flexible, whether it was woody, how tall would the plant grow, and based on how it appeared externally, he classified them into herbs, shrubs, and trees. Herbs obviously are the shortest of the three. It has kind of like a somewhat flexible stem, not too woody. It does not produce too many branches, and it has a short lifespan in most cases. That's how herbs are. Speaking of shrubs, shrubs are slightly bigger than herbs with respect to their height. They are a little bit more branched than herbs, and also shrubs have stems that are slightly more woodier than herbs. Speaking of trees, trees are some of the tallest kind of plants. Compared to shrubs, they're much taller. There also you see branching, here also you see branching, but the branching in trees does not start from the bottom, from towards the surface of the soil. There is a solid trunk that exists beyond which you can see branching. So visually they are very distinct from each other, which is why he used these criteria for classifying plants. Speaking of classification of animals by Aristotle, he classified animals based on just one feature, the color of blood. If an animal's body had red colored blood, then he called it anema. Anema. Like us and most other vertebrates, of course, you would have eaten a fish, you would have eaten a bird like a chicken, you would have eaten meat of another mammal like a goat, if at all. You are a non-vegetarian, you know that most vertebrates do have blood that is red in color. So that was one group, and animals that did not have red blood, they were grouped in another category called Anema, which did not have red blood. You may have noticed how cockroaches don't have red blood. If you've ever attempted to kill a cockroach, you know that it does not bleed red color blood, it's more or less colorless. So yeah, so this was his basis for classifying animals based on the presence or absence of red colored blood. Okay, now moving on. Further, what was mostly accepted for a really long time was the classification system of Linnaeus, the same Linnaeus that we've learnt about in plant kingdom. Carolus Linnaeus, who's called the father of taxonomy, he proposed this two kingdom system of classification where all of the organisms that were described and discovered up until that point were grouped into two distinct categories. Back then, microscopy had not advanced so much, so they did not really know about what cells are and what kind of structures you can find within cells. They were not aware of what prokaryotes and eukaryotes are. They didn't kind of know that cell wall is present in some organisms' cells, and some of them lack a cell wall and all of that. So when Linnaeus classified living organisms into these two groups, he mostly thought about whether that organism has the ability to move or not. If it could move around like most animals do, then it was considered to be an animal. And if organisms could not move around like plants, obviously, then it would be put under kingdom Plantae. Okay. So in this two kingdom system of classification, you have one kingdom Plantae and one kingdom Animalia. For a very long period of time, even after Linnaeus passed, this system was used for classifying living organisms. But as time passed, as science progressed, as technology evolved, developed, people started getting to know more and more features about plants and animals. Eventually, they figured that plant cells have cell walls, animal cells don't have cell walls. Remember cell theory? So that is when people started discovering specific cellular aspects of these living organisms. As and when that happened, they started to include more criteria for classification here. If it has a cell wall, you call it a plant. If it doesn't have a cell wall, then you call it an animal. And they added additional criteria. But despite that, there were some drawbacks with the two kingdom system of classification. Of course, there are some advantages as well. One major advantage is you don't have to go on and explain to people what is the basis for classification, because it's very simple. It's one of two things. Either you're looking at whether the animal can move, whether the organism can move around or not, or you're looking at whether the cells of the organism has a cell wall or not. These are very easy to understand, and it's also easy to classify, right? So there's no too much breaking of head trying to decide where a particular organism has to fit, right? So this was simple, and it was easy not just to classify, but it was also easy to understand. But there were some problems. Let's see what the disadvantages are. There are two major disadvantages. The first one is that this two kingdom system of classification did not differentiate between a lot of factors that had come into light with advancing technology. Okay. So people started noticing that in some cells there was a membrane surrounding the genetic material, while some cells did not have. They classified cells based on whether nuclear membrane is present or absent, okay, by that time. So here what happened was, since Linnaeus considered very few characters or criteria for classification, in the system of classification, organisms that were prokaryotic, which did not have a nuclear membrane, were put together in the same kingdom which had eukaryotic cells that contained a nuclear membrane. For example, bacteria and cyanobacteria, these are prokaryotic. They were grouped along with higher plants such as algae, pteridophytes, gymnosperms, and most other plants which were eukaryotic in the same kingdom, Plantae. They are very different structurally. The cells are very different structurally. Regardless of that, they grouped them in the same kingdom, Plantae, just because they could not move or just because they had cell walls. Okay. The second thing that they kind of ignored and did not distinguish between was whether the organism was made up of a single cell or whether it had multiple cells. Okay. So it did not distinguish between unicellular and multicellular organisms. You know, Chlamydomonas is a unicellular green alga. Unicellular Whereas Pyrogyra is also an alga. This is multicellular. All of these, like Chlamydomonas, Pyrogyra, were included under the group Algae, which had other multicellular photosynthetic organisms as well. All of them were grouped under the kingdom Plantae, which is also not okay, right? Next, they did not check for the mode of nutrition of those organisms. It was not considered at all. They put together photosynthetic or autotrophic organisms along with heterotrophic organisms in the same group called Plantae. Can you imagine fungi along with algae in the same group? They're so distinct from each other. Even if you look at their cell walls, plant cell walls are made up of what is called cellulose mostly, and fungal cell walls are made up of chitin. Even their cell wall composition is very different from each other. The structure, body structure is different from each other. Their modes of nutrition are different from each other. They do not pay attention to any of these details and just, very conveniently put them into the same kingdom, Plantae. So, like we know, fungi, funny guys, they are heterotrophic, and green plants, they are autotrophic. They vary in their modes of nutrition, but despite that, they were grouped under the same kingdom. Fungi are known to have a cell wall that is made up of chitin, while plants have a cell wall that is mostly made up of cellulose. This was also a difference that was not considered in the two kingdom system of classification. This is one thing. Secondly, there were many organisms that couldn't quite fit into either plant kingdom or animal kingdom. For example, Euglena. Euglena has a Euglena suffers from an identity crisis. It can't decide whether it wants to be like a plant or it wants to be like an animal. We call that a mixotroph. How are plants and animals different from each other? Plants know how to make their own food. We depend on them for food. But amoeba is a special kind of feature, a special kind of creature which has features from both plants and animals. It does have chloroplasts like plants, but it can also engulf other organisms and feed on them like animals. Okay, so whether an amoeba behaves, not an amoeba, a euglena. Did I say amoeba since then? I meant euglena. I'm sorry if I mentioned amoeba. Strike it out in your notes and write it as euglena. Okay, so if a euglena has sunlight available, then it behaves like a plant, performs photosynthesis, it has similar pigments like higher plants, it has chloroplasts and all of that. But in the absence of light, it behaves like an animal and feeds on other smaller organisms. Okay, so where do you put that? It is showing photosynthesis sometimes, sometimes it is not. So is it a plant, is it an animal? That is a difficult kind of organism to classify in a two kingdom system of classification. So are slime molds. Remember I told you every time they classified organisms into one of these two kingdoms, they looked for whether it had a cell wall or not. Now in the life cycle of slime molds, there is one phase where cell wall is absent and there's one phase where cell wall is present. Now where do you put this organism? So these were confusing. So by then, because science had advanced, biology research had advanced, microscopy had advanced, people started getting to know more and more details, and it became difficult for people to put organisms into just one of these two boxes. Okay, so that is where there was a need to kind of consider more and more criteria for classifying living organisms, and they started using, apart from morphology, of course morphology is very important, but just apart other than gross morphology, they started using other characteristics as well for classifying living organisms, such as anatomy. Yeah. What is the wall made up of? Chitin, peptidoglycan, cellulose. What is it made up of? Mode of nutrition. Is it autotrophic, heterotrophic? Where does that organism live? Is it in land? Is it on land? Is it in water? Is it in salt water? Is it in fresh water? Where does it live? That is something that they considered. And then how do they reproduce? Do they reproduce asexually? If yes, how? If they reproduce sexually, how do they reproduce sexually? How are the evolutionary relationships among organisms? All of that they started to consider while classifying living organisms. You might wonder why evolutionary relationships are important. I've mentioned this earlier as well when we discussed the living world. It's important because organisms that have evolved from the common ancestral line will have more similarities, and they have to be put into similar categories, similar taxonomic categories, because there are more shared similarities. sometimes by appearance, by just looking at the external appearance, you might not be able to kind of, deduce whether it belongs, it should be in the same category or a different category. I gave you this example in the previous chapter as well. I think I mentioned how dolphins look more similar to sharks than they do to us. But if you look at our evolutionary, lineage, then you will get to know that we are more closely related, right? And also, bats look more similar to birds than they do look to mammals. But still, bats are mammals, right? So it's important that you study the evolutionary history. That way you will have a more accurate way of classification. All right. Now that two kingdom system of classification was adequate for all of the reasons that I mentioned, there was a need for a new classification system that considered whether the cells were prokaryotic, eukaryotic, unicellular, multicellular, what is their mode of nutrition, how are they undergoing reproduction, and all of that. So much later came Whittaker's, R.H. Whittaker's five kingdom system of classification. So here in R.H. Whittaker is name of the person. He proposed a five kingdom system of classification. Unlike Linnaeus, he used many criteria for classifying all living organisms into five different groups. Imagine there are as many as 1.7 to 1.8 million species on our planet. All of these have to be fit into these five kingdoms. Okay, so it's a big job. So you will need more and more criteria for classification. So, what are some of the criteria that he used? Let's take a look at that. He checked for the cell structure, whether the cells are prokaryotic or are they eukaryotic. Next, body organization. Whether that organism was made up of just one cell, in which case we call it unicellular, or was it composed of many cells, more than one cells, which is called multicellular. Then you have mode of nutrition, whether that organism can synthesize food by itself or does it depend on other organisms to synthesize food, autotrophic or heterotrophic. In case it is heterotrophic, is it showing absorptive heterotrophy or ingestive heterotrophy? What is absorptive heterotrophy, ingestive heterotrophy? I'll tell you shortly. Okay, don't worry too much about that. Now, the next criterion is reproduction. If it undergoes asexual reproduction, what are some ways in which it undergoes asexual reproduction? And if at all it undergoes sexual reproduction, how exactly does that happen? And finally, phylogenetic relationships. Phylogenetic relationships is nothing but evolutionary history or evolutionary relationships. Okay? Now, let's see exactly how Whittaker classified living organisms into five kingdoms. So let's start off with all of the living organisms that have been described so far. Have so many living organisms. How many? A lot. In millions. That many species have been discovered. Now, the first thing that we are going to check for is whether that particular organism, a prokaryote or is it a eukaryote? Of course, when you're considering all living organisms, some of them will be prokaryotic and a lot of them could be eukaryotic, right? So, all of the prokaryotic organisms of the 1.7 to 1.8 million will be grouped under kingdom Monera. What about the remaining kingdoms? This is the first kingdom. What are we left with now? Among the 1.7 to 1.8 million species, we've grouped all the prokaryotes in Monera. What are we left with? We're left with eukaryotes. We are going to ask the next question to eukaryotes. Are you unicellular or are you multicellular? Is it made up of a single cell or is it made up of multiple cells? If it is unicellular, it is grouped within the next kingdom, which is called kingdom Protista. What are we left with? We are left with multicellular organisms. What is the next question we're going to ask? We will check for its mode of nutrition. Is it autotrophic or is it heterotrophic? Or heterotrophic? If it has a superpower and can synthesize food by itself, then you call it autotrophic. If it is autotrophic, then obviously you might have already guessed it is included under kingdom Plantae. It's also called the plant kingdom. Okay. Then what are we left with? The remaining heterotrophic organisms. Now we're going to ask if it shows absorptive heterotrophy or ingestive heterotrophy. First, let's understand what is absorptive heterotrophy and ingestive heterotrophy. Why did I get a green screen? Minute while I switch. Okay. So, we are speaking of the difference between absorptive and ingestive heterotrophy. Both of them are types of heterotrophy, which means both organisms cannot prepare food. They are dependent on other organisms for nourishment. In absorptive heterotrophy, let's take the example of fungi. You know how fungi are, right? They are said to show saprotrophic nutrition. What does that mean? Let's say this is a log of wood, or let's say a log of wood would be, okay, so let's say this is the floor or the soil, whatever. You know how fungi are ubiquitous, they are found everywhere around us. On the same soil, there is a dead leaf. Let's just say there is one animal tissue as well. Some animal, dead animal's bone is also there. So fungi that are growing here, what color do you think I should represent fungi in? Let's use blue color. Okay. So let's say there's fungi growing here. Obviously, it will not be so big. Just for your reference, I'm drawing like big cells here. So these are fungi growing and these are fungal cells. If a fungal cell or a fungus body has to obtain nourishment from outside, it does not have a mouth. It does not have an internal cavity in its body like animals do. So it can't literally eat food and obtain nourishment from them. So what does it do? Similar to how we have enzymes in our digestive tract, like we have enzymes that are produced by our salivary glands, there are enzymes that are produced by our stomach like pepsinogen, there are enzymes that are produced by our pancreas, enzymes produced by our small intestine. All of that collectively will start acting upon the food that we eat. It will break it down and the broken down components are absorbed by our blood and transported to our body, right? Everywhere across the body to every one of our living cells. So here what happens is, they don't have like an organ system level of organization. They don't even have an organ level of organization, fungi. So every cell takes care of its own need. So if this cell requires food, it requires nourishment, it draws it from the surroundings. It's not like that cell has a mouth, it will go eat it. Okay. So what it does is, unlike us, where enzymes are secreted inside the body, in these fungi, enzymes definitely will get produced. But those enzymes, once it is produced inside the cell, they will be released outside. Okay. So these enzymes will start breaking down the tissues present. It will break down the bones, it will break down the leaf, whatever organic matter is there, it will break down. Once that is broken down, the fungi will absorb the nutrients. So that is called absorptive heterotrophy. How do you think our bodies decompose once we die? It gets decomposed because of fungi like this, like these, because of saprotrophs, because of absorptive heterotrophy. So they can't literally eat us. They will secrete enzymes outside. Those enzymes will break us down, and the broken down components are taken into the cells of the fungi. That is absorptive heterotrophy. As opposed to that, we have ingestive heterotrophy, which is found in animals. Now, this is also called holozoic nutrition. Here what happens? Let's take an example of us. We have, okay, so let me draw one funny human face. This is the nose, this is the mouth, this is the eyes. Let's put one juttu like this. Okay, okay. This is the body of a human. I haven't drawn a human body in a really long time. Don't judge my diagram, okay? So this is the esophagus and stuff. And where do we have stomach? J-shaped organ. Then you have small intestine, okay? And then you have the large intestine. And then beard, rectum, anus, and all of that, right? This is the human body. Cool. So here what happens is we eat the food. We literally eat food. We eat food. Okay, so this eating of food is called ingestion. We literally take food into our body, not just us, every animal from porifera to mammals. You will see that inside the body of an animal, there will be a body cavity. You've seen that spongocoelent sponges and all of that. That's where the food will go, right? So there is an internal body cavity. That is where the enzymes are released. So after you eat this apple or whatever food you ate, okay, so in different parts of the alimentary canal, different enzymes are secreted. The carbohydrates in the apple get broken down. Whatever components are there in the apple, it gets broken down. It gets absorbed by your large intestine, it gets absorbed by your small intestine. And then it is distributed or circulated or transported throughout the body, right? So for us, for obtaining nourishment, you can't keep an apple on your skin and expect your body to absorb it like fungi do. It has to go inside the body. We have to eat it. Eating is ingestion, not injection, ingestion. Okay. So once it goes inside the body in different parts of your alimentary canal, like I mentioned, for at least 10 times in the past, it will get broken down and then it gets absorbed. And that whole story is ingestive heterotrophy. In both cases, neither are animals nor are fungi capable of making food on their own. They are dependent on other organisms. It could be plants or animals. Okay. So yeah, that's heterotrophy and the two types of heterotrophy. In case it shows absorptive heterotrophy, then obviously you would know, I already gave you examples. It will be included under the kingdom fungi. If they show ingestive heterotrophy, then it is included under kingdom Animalia. So here it is clear how we arrived at 5 different kingdoms. Okay, so here you have kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. If you look at these criteria that I mentioned earlier, we've covered these 3 criteria. About reproduction, phylogenetic relationships, we haven't yet spoken. At the kingdom level of classification, we will not really pay too much attention to how reproduction happens or phylogenetic relationships are. You know, this is not the end, right? Classification happens at multiple levels. After kingdom, you have phylum. After phylum, you have class and all of that. So during further classification, these criteria will be used. When we're discussing kingdom fungi, you will realize how reproduction can be used as a criterion for further classification of organisms, okay? Now, kingdom fungi is also called kingdom mycota. Keep that in mind. If you hear the term mycota, that is about fungi. Okay? Now, yeah. So this is the table that NCERT has given. In the first two rows, it's very obvious. Monera is prokaryotic. If the reason it's prokaryotic is because it does not have a nuclear membrane. Nuclear membrane is absent, that's why it's prokaryotic. In the remaining ones, nuclear membrane is there, they're eukaryotic. Okay. Next, body organization. Monera and Protista includes unicellular organisms. The remaining three kingdoms, fungi, Plantae and Animalia, are mostly multicellular. Next, cell wall composition. In Monera, some of them have cell walls, some of them don't have cell walls. Same goes for Protista. Some organisms have cell walls, some organisms don't have cell wall. In fungi, all of them have cell walls. In plants, all of them have cell walls. In animals, none of them have cell walls. Okay? Then, mode of nutrition. Since mode of nutrition was a criterion that we used only for the classification of fungi, Plantae and Animalia, over here, mode of nutrition is fixed. In fungi, it is heterotrophic, absorptive heterotrophy or saprotrophism. In animals, it is heterotrophic, it is ingestive heterotrophy or holozoic nutrition. In plants, majority cases, they are autotrophic. Okay. But since that was not a criterion used during classification of Monera and Protista, here you have a mixed bag of different kinds of organisms showing different modes of nutrition. So here you can find autotrophic, heterotrophic, similar to fungi, heterotrophic, similar to animals, all of that you can find in these two groups. All right. So now let's talk about another system of classification, which is called the six kingdom system of classification, or the three domain system of classification. There was one person called Carl Woese. He studied bacteria extensively. When he started studying bacteria or prokaryotic organisms, he realized that bacteria have two different groups. One is called Archaebacteria and the other one is called Eubacteria. Okay. So what he did was he studied them in detail and he figured the cell walls of Archaebacteria were very different from the cell walls of Eubacteria. A type of RNA called 16S rRNA was also very different in both Archaebacteria and Eubacteria. Both of them are prokaryotic. Both of them were grouped under Monera. But after looking at the differences in structure and structure of RNA between these two groups of bacteria, he decided to split kingdom Monera into two different kingdoms. He split kingdom Monera into kingdom Archaebacteria and kingdom Eubacteria. Okay. So from 5 kingdoms, we have 6 kingdoms. The other 4 kingdoms, eukaryotic kingdoms, they remain unchanged. Change has happened only in Monera. Monera no longer exists, but instead of Monera, you have 2 other kingdoms which are derived from Monera, Archaebacteria and Eubacteria. And why? What is the basis for their classification? It is based on their cell wall structure and also 16S ribosomal RNA differences. Okay. Not only does he classify organisms into 6 kingdoms, he groups them into 3 major domains. One domain includes all of the eukaryotic kingdoms, which is called domain Eukarya because it includes all eukaryotic kingdoms. And you have one domain for Archaea, one domain for Eubacteria. Okay, so Archaebacteria are included under domain Archaea, Eubacteria are included under the domain Eubacteria. Okay, bacteria. So these are the 3 domains, these are the 6 kingdoms, keep that in mind. Now let's move on and learn about each of these 5 kingdoms. In this chapter, we're going to learn in detail about kingdom Monera, Protista, fungi, Plantae and Animalia. Very briefly we will touch upon because you have 2 entire chapters dedicated to Plantae and Animalia. There's a chapter called Plant Kingdom which I will teach you next. There's another chapter called Animal Kingdom which you may have already dealt with in zoology. So over there you will exclusively learn about the further classification of those kingdoms. So here we're mostly learning about Monera, Protista and fungi. Alright, so let's start off with kingdom Monera. Now what do we already know about kingdom Monera? When we were classifying, I mentioned how Monera includes only prokaryotic organisms, right? So prokaryotic organisms means obviously they're only bacteria. So the only organisms that you can find in Monera are bacteria. So it's said that bacteria are the sole members of kingdom Monera. Over here, sole does not mean the soles of your feet. Soul does not mean the soul that leaves your body when you realize your crush does not like you back. It's not that soul. Soul here means only or exclusively. Okay, so it means bacteria are the only members that you can find in kingdom Monera. Now, it's also mentioned that these are the most abundant microorganisms. What are microorganisms? Microorganisms are those organisms that are like too tiny. You can't see them with your naked eyes. You can't see them like you see a tulsi plant or a mosquito or a banyan tree. You can't see them at all unless you're looking at them under the microscope. They're that tiny. All such organisms which cannot be seen through your naked eye are called microorganisms, and it includes viruses, many different types of algae, many different types of protozoans, and I don't know why I look up when I think, but yeah. So, yeah, and bacteria too. So that among all of those microorganisms, bacteria are the most abundant meaning most maximum in number. Okay, so if you take like a handful of grain of sand, of course you will be able to find many different types of microbes there. Many different species of microbes. You may come across certain protozoans, you may come across certain fungi and bacteria, but among them all, the number of bacterial species will be the highest. That's what they mean. Look everywhere, in the air, in water, on land, anywhere. Microbes exist everywhere, but among the microbes, bacteria are in the highest numbers. Okay? Yeah. And some of these bacteria are crazy. Okay? They live in some extreme climatic conditions. Like, I'm saying extreme because that's extreme for us. For them, it's like normal. It's like a chill spa day. But they live in regions where if we go, no, our bones would melt. Okay, even in such conditions, bacteria are known to not just survive, they thrive there. They live very happily like kings there. So some of them grow in conditions where the temperature could exceed 80 degrees Celsius. Okay, in summer in India, if it exceeds 40 degrees Celsius, we're all panting and people die of heat shock, right? So, but these guys, they survive there. In hot springs where the temperature could exceed 80 degrees Celsius, even in deserts where there is very little water, in snow where the temperature is sub-zero, like very low, and in deep oceans where the pressure crazy high. So these are extremes for us. We cannot survive there. Most organisms cannot survive there. But bacteria have come up with mechanisms which ensure that they can survive there as well. Okay? Now, some of them live in or on other organisms. In means inside the body. On means on top of the body of organisms. As parasites, that means they are heterotrophic. They depend on the organism that they're living in or on for their nourishment. Okay, you may have seen how many bacteria causes diseases, right? Cholera is caused by bacteria, and typhoid and tuberculosis. All of these are bacterial diseases. What do these bacteria do inside of our body? They live like parasites. They draw nourishment from us. They are parasitic, they are pathogenic, and some of these bacteria cause skin infections also. You all are teenagers, you may have seen people, or you may have experienced having acne on your faces. Sometimes the acne also has bacterial growth. It's living on top of your skin, right? Not just in humans, even on other organisms, other plants and animals. You can find bacteria living inside the body or outside the body and drawing nourishment from that particular organism, in which case we call it parasites. If it lives inside the body of another organism, then you call it an ectoparasite. If it lives on or outside the body of another organism, then you call it endoparasite. It's a single word. Okay, one single term. All right. Now, let's speak of the shapes in which you can find bacteria. We've spoken about this in the chapter Cell the Unit of Life as well. There are many different shapes, of which there are 4 major shapes. Either bacteria can appear spherical, in which case we call them cocci. You've heard of streptococci, staphylococci, right? Next we have bacilli like lactobacillus, Bacillus thuringiensis. All of those are rod-shaped bacteria. And then we have helical bacteria like spring-like coiled that are called spirilla or spirillum is singular. Then we have comma-shaped bacteria. It appears comma-shaped because, it almost always has like a very long tail that allows it to move. So that gives it like a comma-like appearance. For example, you've heard of Vibrio cholerae that causes cholera, right? So that is also comma-shaped, okay? So these are some shapes that you can find in bacteria. Now let's speak of the various modes of nutrition we can find in bacteria. You know how structurally they are very simple, right? They're prokaryotic, they don't have a complex cellular organization. They just have very few organelles like ribosomes and a very simple sort of structure. But in their behavior, they can be pretty complex. And these are organisms that display the most extensive metabolic diversity. What do we mean when we say it's extensive metabolic diversity? Metabolic diversity means various modes of nutrition. Okay. So when we speak of plants, we know that majority of them are autotrophic. When you speak of animals and fungi, you know that they are heterotrophic. Over there, we have specifically used mode of nutrition as a criterion for classification, but we didn't do that for Monera, right? So here you can come across organisms that are autotrophic, some of them are heterotrophic. Even under autotrophic bacteria, there is further more different types. Some of them are photoautotrophic like most plants and some of them are chemoautotrophic. So let's learn about that. So here in bacteria, you can find how some are autotrophic. Auto means self. They are self-sufficient. They can synthesize food by themselves. And B, heterotrophic means they depend on other organisms for nourishment. First, let's look at autotrophic organisms. So here, there are two types again. There's photoautotrophism and chemoautotrophism. Photo itself means light, right? So photon is about light. So if organism uses sunlight, traps the sun's energy, uses it in the synthesis of ATP, uses that ATP to synthesize glucose, then you call it photoautotrophism. Chemoautotrophic organisms, as the name itself indicates, chemo means chemical. So what they basically do is they will oxidize certain inorganic compounds. They will perform certain chemical reactions because of which ATP gets generated and they use that ATP to synthesize glucose. Okay. So photoautotrophic mode of nutrition you can find in plants, you can find in some protists, you can find here in bacteria also. But chemoautotrophism is something that you can find mostly in bacteria. Okay. So this is only in bacteria. Which one? Chemo. Photo is found across different kingdoms, but chemoautotrophic nutrition is found only in bacteria. Now, speaking of heterotrophic nutrition, it just means that they themselves cannot synthesize food. They depend on other organisms. Depending upon whether the other organism is living or dead or whether it is a mutualistic relationship, you have different types. For example, if this depends on another organism that is living, then you call it the host. In that case, we call it a parasite, like I told you earlier. In case it is dependent on another dead organism for its nourishment, then you call it saprophytic. If it depends on another organism and in turn helps out the other organism, it's not like draining its energy away, it's not like killing that organism by parasitizing it. If both of them are benefited from that interaction, then you call it symbiotic association, where both are benefited. All of these kinds you can find in bacteria. Okay. Now, within Monera, you have two distinct groups of bacteria. The first one is called archaebacteria. Archae. So when somebody is talking like one Ajit Tata, like one grandfather, you say you are archaic. You're sounding so archaic. You're sounding like you're from 1900s, right? So archaic just means that it's very, very old, ancient. That's what it means. So these archaebacteria are also believed to be like very, very ancient bacteria, which means that they've lived on our planet for billions of years. They've existed on our planet for billions of years. And these archaebacteria's characteristic feature is that they live in habitats which are considered hostile to humans. Not humans, to most other organisms. Most other organisms cannot survive in such conditions. These guys will be like, oh, bro, we got this, we can do this, and they will go chill there, they survive there. But the thing that we must understand here is, we are calling them extremophiles because those conditions are extreme for us, not for them. If you take an archaebacterium that is living at 100 degrees Celsius and put it in your living room, it will not be able to survive. So what is normal to us is extreme for them. We are calling them extremophiles, keeping ourselves at the center. Okay, for us it is extreme, they can survive in those conditions. That's what it means. Okay, for them it is very normal. For them, how we live is extreme. Okay, so yeah, these are archaebacteria. Over here, you have to learn about 3 different categories. You have halophiles, thermoacidophiles, and methanogens. What are halophiles? Halo is not the halo around the head of gods. Halo here refers to salinity. Okay? So, halo means salinity. Philes or philia means love for something. Okay, so on your Instagram bio, some of you will put your selenophile and bibliophile. I like all of those fancy terms, no? So philia basically means love for something. So these guys are called halophiles because they apparently love salt so much that they are found in extremely salty habitats, so found in extreme salt environments. And then we have thermoacidophiles. Thermo relates to temperature, acido relates to pH or acidity, philia again is love for something. So these guys love to thrive at high temperature and low pH. Temperature has to be very high as 80 degrees Celsius or beyond that. pH could be as low as 2, which is like highly acidic. Under such conditions, they grow very well. So they are called thermoacidophiles. Then we have methanogens. Methano relates to ethane, ethane. Gens relates to generating. So these are bacteria that have the ability to generate methane, which is inflammable. It is used in the production of a biofuel called biogas. So if it produces methane, so what? What is so extreme about that, you might think? What is extreme about these bacteria is that they can survive without oxygen. We can't, last an hour or probably a couple of minutes without oxygen, but these organisms can survive very well without oxygen. So they are anaerobic. That is quite extreme, isn't it? Then we have, where do you find them? You find them in marshy areas. Marshy areas are where there is waterlogged conditions. In the soil, there's not sufficient aeration, so they're not having much access to air or oxygen. And they also are known to live inside the digestive system of ruminant animals like cows, buffaloes, goats, sheep, and all of that. So they can live in very dark, anoxygenic conditions. They are anaerobic organisms. So yeah, that is quite extreme if you ask me. So I've been saying that most organisms cannot survive in such conditions, but these guys, they thrive there, they're very happy there, and all of that. How is it that these organisms can survive in conditions where most others can't? They are capable of surviving there because of their modified cell wall structure. Okay, so these bacteria cell walls are slightly different from the cell walls of other eubacteria. These archaebacteria cell wall is made up of a substance called peptidoglycan is regular bacteria. This is called pseudopeptidoglycan. Or pseudomurine. Okay, that's what it is made up of, which will allow it to withstand such harsh conditions. Now, let's speak of eubacteria. I told you, if you consider all of the microbes in the world, the most abundant ones are the bacteria. Now, within bacteria, you have archaebacteria, eubacteria. Now, if you're comparing archaebacteria and eubacterial species, more abundant one is the eubacteria. Okay, so there are thousands of different species of eubacteria, which are also called true bacteria. U means true. I told you this when we discussed prokaryotic, eukaryotic as well. U means true. Okay, so that's why these are called eubacteria. Now, these are characterized by the presence of a rigid cell wall that is made up of a substance called peptidoglycan. It has polysaccharide, it has amino acids, short peptides joining the polysaccharide. So it's called a peptidoglycan. Glycan is sugar. Peptido, amino acids, short chains, peptides, that is peptido. Okay. Now, sometimes these eubacteria are motile, which means they can move around, in which case they will contain what is called a flagellum. Flagellum allows it to move around, so it is a locomotory structure. Only if the bacteria is motile, then it will contain flagellum. Remember, I told you how most species of Vibrio bacteria have a long tail. By the tail, I meant they have a flagellum only that will help it move around. That will keep spinning and then help the organism propel forward. Okay. So, if the bacteria are motile, then flagella will be there. Now, these eubacteria are of two kinds when it comes to their mode of nutrition. Some of them are autotrophic, a lot of them are heterotrophic. Okay. So, autotrophic, few of them, heterotrophic, most of them. Again, under autotrophic, you have photosynthetic bacteria and chemosynthetic bacteria. Some examples for photosynthetic or photoautotrophic eubacteria are cyanobacteria. Cyanobacteria are nothing but blue-green algae. Remember, I told you in the beginning of the chapter that back in the day, there was a time when there was only two kingdom system of classification. You either put organisms in kingdom Animalia or you put them under kingdom Plantae. At that time, when they discovered something like a cyanobacterium, which was prokaryotic, which could photosynthesize, they had automatically put that under kingdom Plantae. Since it was considered a plant, they assumed that it was like algae because it was photosynthesizing, right? So back then, because it had a bluish green color because of the pigments, the kind of pigments it had, it was called blue-green algae. Later, they realized that it does not have a nuclear membrane around it, so it's prokaryotic, it should be moved to Monera, and Monera has only bacteria. They changed the name of blue-green algae to cyanobacteria. Today, many people still use blue-green algae, even if you're in your question papers, blue-green algae is given, then just know that it is cyanobacteria. In fact, the color cyan means blue-green. Cyan is a color. Why do we call this cyanobacteria? Cyan is a color. It's a blue-green color, something like this, turquoise kind of blue-green color. So that is cyanobacteria. Okay. So if they are photosynthetic, that means obviously they should have a mechanism to trap sun's energy, right? Unless they have pigments, how can they capture the sun's energy? They can't. So these photosynthetic bacteria will definitely have pigments such as chlorophyll A found in higher plants also. And these photosynthetic bacteria could be unicellular, in which case they are made up of a single cell. They could be colonial, where many cells are coming together forming a colony, or they can be filamentous. Filamentous means cells lined end to end like this, and it forms a long thread-like structure that is called filamentous. What you're looking at here is a filamentous blue-green alga called nostoc. This is a very good example for it. Okay, where is it? Okay, apart from nostoc, you have another example called Annabena as well. There are some very amazing features about blue-green algae that I want you all to know. Don't just read this for the sake of your exams. You also want to develop an interest about the kind of amazing diversity you see in life around you. And yeah, these are very cool organisms. They do some really great stuff and they have some amazing superpowers about which we will talk right now. Okay. So where do you find them? Since they are very, very small, they don't have roots to absorb water, they are photosynthetic. Obviously, they will be found in regions where water is sufficiently available. So they are found in water. It could be freshwater or marine habitats. But if in case they are found on land, they will be found on land only where there is moisture. They cannot grow in dry habitats because they are photosynthetic, they need water. Okay. Now, you can notice how on the outer surface of these blue-green algae, there is what seems like, what color should I use? Orange. Always. Yeah. So there is an outer jelly-like sheath that you can see around them. This is literally jelly-like. It's a mucilaginous sheath. You can call it a gelatinous sheath. In the diagram in NCERT, they've mentioned mucilaginous sheath. In the description part, they've written gelatinous sheath. Both mean the same thing because it is mucilaginous, made up of mucus. It has a jelly-like consistency. Okay. So this will be present around most of them. Okay. So what is the importance of this mucilaginous sheath? Let's understand. It does three things. Okay. Even in water bodies where they are growing, though these are very, very small, these can act like food for slightly smaller animals like amoebae or certain zooplanktons that are also living in water. They feed on them like food. Okay. If it has like a slippery nature, it is a little difficult for other animals to engulf it. That way it is getting protected. Secondly, like these are also like plants, they require mineral nutrients to survive, to perform photosynthesis. Where do they get mineral nutrients from? Plants get it from the soil. There is a root system that will absorb minerals and pass it on to the aerial part of the plant. But these guys are very, very simple. They don't have such elaborate structures. So what do? They absorb nutrients, minerals present from its surroundings. So basically, since these are mostly aquatic, in the water surrounding this blue-green algae, minerals will be dissolved. Okay. If you look at water present in soil or river or any water body, it's not pure water. It's not like distilled 100% pure water. There will be minerals dissolved in it, right? So, if you think of how much concentration the minerals are present in, the concentration will be very less. Considering how much water is there, mineral concentration will be very less. So, for them to absorb them, take up the minerals into their cells, there will be very dilute solution of minerals present around them. So what happens with the mucilaginous sheath is it kind of concentrates the minerals present in the water around it so that the cells can more easily take it up. Okay. So it helps in concentrating minerals. Concentrating is not concentrating. Concentrating is increasing its quantity in its immediate vicinity. Okay. Next, what else does it do? It also kind of helps prevent water loss or desiccation. What is desiccation? Desiccation is loss of water. These require water to function and survive. So let's assume this is a blue-green alga growing in a water body like an ocean or a sea somewhere. Okay. So it's possible that the ocean water, the waves hit the shore and go back. While the waves are tracing back, the algae are left on the shore where there is not sufficient water. So if there is not sufficient water, they will not be able to survive for long. So they will get dehydrated, after which they will not be able to survive. So during that period, while they are resting on the shore, while they are dropped off on the shore, having a jelly will help retain moisture. That's how Vaseline works, right? Why, when it's cold, when your skin is very dry, do you apply moisturizer? What does a moisturizer do to your skin? What does a coating of Vaseline do to your skin? It holds back the moisture, it keeps your skin hydrated. Exactly same thing happens here. We have to artificially apply Vaseline on our skin, moisturizer and body lotions on our skin, but this is born with a inbuilt moisturizer, inbuilt jelly coat that will help it stay hydrated for as long as possible. And let's say after a couple of minutes, another big wave hits the shore and takes this back into the ocean, it's safe. Until then, the mucilaginous sheath will help protect it. Okay. So that's one very important thing about this. Okay, so it has a gelatinous sheath. Another super power that this has is that it has the ability to fix atmospheric nitrogen. I'm drawing specific cells in red color. You can notice how these cells are very different from the other vegetative cells. These cells are called heterocysts. Heteros, already mentioned there, so let me not save myself the trouble. This is a heterocyst. Okay. So what is the superpower that heterocysts have? Heterocysts have an enzyme called nitrogenase. Okay. So this is so cool. You know how plants require nitrogen for their survival and metabolism. They require a lot of nitrogen. Okay. We also require nitrogen. We get that from the food we eat. But these guys have to absorb it from the surrounding medium, right? Nitrogen is important because you require nitrogen to make DNA. DNA has nitrogenous bases. You require nitrogen to make proteins because proteins have amine group which has nitrogen. So nitrogen is very essential if an organism has to grow. So usually in soils, nitrogen is deficient. Even in aquatic environments, nitrogen could be deficient. So what they do is, why it is deficient also I'll tell you. It's not like there is not sufficient nitrogen on our planet. There is a lot of nitrogen present in its molecular form in our atmosphere, but poor thing, these plants and these microbes do not have the ability to utilize that nitrogen. N2 molecular nitrogen is bound. The two atoms of nitrogen are bound by very strong covalent bonds. It's difficult to break it and utilize it for their metabolic reactions. It's very difficult. So they don't usually absorb nitrogen or take in nitrogen in the form of molecular nitrogen N2. Even though it's abundantly available, plants cannot take it up in that form. So plants rely on nitrogenous molecules like ammonia, nitrites, nitrates. Those forms plant can absorb. Here, this organism has special cells called heterocysts that has an enzyme called nitrogenase, and this nitrogenase enzyme is a it's like a master, okay. So it can very efficiently break the covalent bonds between nitrogen molecule, between the nitrogen atoms in a nitrogen molecule and synthesize ammonium. So that ammonium can be utilized by these organisms, okay. So the process by which this atmospheric nitrogen is converted into ammonia, that is called nitrogen fixation, and that can be performed only by these cells because they have this enzyme called nitrogenase, which the other cells do not have, okay. And also for these, for the nitrogenase to be active, to be able to fix nitrogen, anaerobic conditions have to be maintained. Think about it, okay. So specifically in these cells, there are thick walled cells, and they make sure that the region inside the cell where nitrogenase is active, they make sure that anaerobic conditions are maintained. They will make sure that oxygen does not reach that place, and they're so intelligent, they're so clever, they're brainless, they are just a few cells, and still they are capable of so much, right? So that's the beauty of nature. So they have heterocysts that will help in fixing atmospheric nitrogen. Again, when I say fix atmospheric nitrogen, I mean to say that the nitrogen molecule present in the atmosphere will get converted into absorbable forms such as ammonia or ammonium ion, NH3 plus something, okay, poor chemistry. Let me just write it down in words. Ammonia, NH4 is the ammonia, NH3 plus is the ammonium ion, right? So it will convert it into ammonia. NH3, NH4 plus, oh yeah, NH4 plus is ammonium, NH3 is ammonia. I think so. Okay. So one other thing is, these are known to form blooms. Just like every human has a good side and a bad side, all microbes also do good and bad for the atmosphere, for the environment, not atmosphere, environment. So some of these blue-green algae, when there is sufficient nutrients present in wherever it is growing in water bodies, they tend to grow very, very rapidly. They keep on increasing in number. They just spread across the entire surface of the water body in what is called algal blooms. This pollutes the water body and it will affect the health of the water body. See, there are a lot of organisms that are dependent on this water body, right? There will be many fishes living here. There will be many other different types of animals. There will be plants that are growing underneath the water that are submerged. They all are dependent on water. What happens when these algae spread across the surface of water bodies? The plants that are growing underneath will not get sunlight to survive. Plants require sunlight. It's like food for them, right? So this will kind of block the entry of sun into the lower part of the water body. So they will end up becoming deprived of sunlight and they will die. And then when these are growing at a very rapid rate, they will also keep dying at a very rapid rate. And for decomposing these algae, the other microbes that are living there, the decomposers like other fungi and bacteria that are living there, heterotrophic, they will start using up all of the oxygen present in the water body in its dissolved form. See, water body has some dissolved oxygen. Fishes are also dependent on that dissolved oxygen for survival. These microbes also need that dissolved oxygen to break the dead microbes down, right? So if those microbes are utilizing a lot of dissolved oxygen, the fishes will not get sufficient oxygen and they could die. Sometimes these are known to release toxins also that can kill fishes. If any livestock feed on that water, it'll, it could affect its health. Sometimes it changes the smell of the water body, it becomes very bad odor. It gives a very bad odor, very bad taste. The water to look at will look very cloudy and it look bad. So it kind of pollutes the water body. So that's the downside of having too much cyanobacteria. Like they say in Kannada, ati adre amritanu vishane. That means too much of elixir is also like poison. Too much of anything good is also not good. Eventually, it'll turn bad. Okay, so that's about the photosynthetic autotrophs, photosynthetic bacteria. Now let's speak of chemosynthetic bacteria. Now these chemosynthetic bacteria, like I mentioned, will perform chemical reactions. Mostly they will oxidize inorganic substances. By oxidizing inorganic substances, they will generate energy in the form of ATP. And this ATP they utilize to synthesize glucose. In both photosynthesis and chemosynthesis, synthesis of glucose is happening. But what is the source of energy for glucose synthesis? In photoautotrophism, it is sunlight. In chemoautotrophism, it is the energy that they obtain by performing some oxidation reactions. Okay, so these bacteria are also very significant in our ecosystem because they help in mineral cycling, like nitrogen, phosphorus, iron. All of these are cycled between different components of our environment, okay, because of chemoautotrophic mode of nutrition. So that is about autotrophic bacteria. Now let's speak of heterotrophic bacteria. There's a lot of variety even over here. These kind of bacteria are the most abundant. Think about it. Among all microbes, bacteria most abundant. Among Archaebacteria, Eubacteria is most abundant. Among eubacteria, you have autotrophic, heterotrophic. Heterotrophic is most abundant. Okay. So these are important decomposers. So these bacteria are also beneficial. Some of them cause harm as well. What are some of the benefits of these bacteria? They help us in making curd, cheese, all these dairy products is because of fermentation by bacteria. They produce many different types of antibiotics, which are medicines or drugs that are used to treat bacterial infections. And they're also involved in fixing nitrogen in leguminous plants. Fixing nitrogen, what is that? I have already told you. There is a bacterium called Rhizobium, which you may have heard about. So this Rhizobium specifically lives in the roots of these leguminous plants. Okay. So legume roots, legume is what? Legume is bean family, beans, peas, all of that are legumes. So if this is like the plant, then in the roots, these bacteria will survive. Okay, so what color? Yellow color. These bacteria will make nodules here and they will survive here and they will end up bringing about nitrogen fixation. Okay. So this is a kind of symbiotic relationship. Previously, I told you how bacteria can show mutualistic relationship between with other organisms. Here, the bacteria helps the plant in fixing nitrogen, provides nitrogen to the plant. In return, the plant gives the bacteria food and shelter. So this is like a mutualistic relationship. Okay. Now, speaking of the downside of heterotrophic bacteria, some of them are pathogenic, which means it can cause diseases, not just to humans, but also to crop plants, to farm animals, and even pets. Some examples of bacterial diseases that you can find in humans are cholera, typhoid, and tetanus. Okay, these are infections that you see in humans. It also causes infection in plants, okay, such as lemon, oranges. You may have noticed sometimes how on top of lemons, some lemons, okay, so if this is like a lemon, you will notice how sometimes there will be, what have I done? There will be what? Some black sort of spots, right? Not like really black, some dark colored spots will be there. Okay, that is called citrus canker. That is caused by a bacterium called Xanthomonas citri, I think. Yeah, so that causes diseases in plants as well as animals as well. So that's about the heterotrophic bacteria. Now, speaking of how bacteria reproduce, bacteria are very, very primitive organisms. Their reproduction is also very primitive. So mostly they undergo asexual reproduction by a process called binary fission. Over here, bi means two. Fission means splitting. So when one bacterial cell becomes bigger, elongates, makes an extra copy of its DNA, splits into two, you get two cells from one cell. That's why it's called binary fission. This happens when the conditions for bacterial growth are favorable. That means there's sufficient amount of food available, there is sufficient space available, temperature is in the optimum range. In such cases, bacteria will want to spread across rapidly, increase in number, so they will go on undergoing binary fission and increase in number. What happens when conditions are unfavorable is they will undergo reproduction asexually by a process called sporulation. They will produce spores that have very thick walls. Remember, these are produced during unfavorable conditions. Whenever conditions are not favorable, whenever it's too hot outside, whenever it is too cold outside, what do? You lock yourself inside your house. You don't step out so much, right? Same thing here. It will build a very thick wall around itself. That thick wall will protect the internal contents, the protoplasm, from extreme heat, extreme cold temperature, or too much, let's say, no availability of water, there is no food, whatever could be the reason for unfavorable condition, whatever that is, this will stay protected because it has a very thick wall. And eventually when the favorable conditions return, these spores will produce new bacteria. Okay, so spore formation happens when the conditions are unfavorable. Do they reproduce sexually at all? Of course, they do undergo sexual reproduction, but that's unlike any other type of sexual reproduction you've heard about. Usually, the moment we say sexual reproduction, we tend to think about formation of male gamete, male sex organ, female sex organ, gametes fuse, form the zygote and all of that. So much drama does not happen in them. They do not have the energy for all of that. So a very, very simple, primitive type of sexual reproduction happens here. So what they do here is these bacteria. See, one of the characteristic features of sexual reproduction is that it will bring about variation, right? So newer genetic combinations, newer genes, newer DNA combinations will arise. Here, DNA being transferred from one bacteria to another bacteria itself is considered as sexual reproduction. Here, gamete formation, gamete fusion, none of that will happen, okay? So over here, there is a very primitive type of sexual reproduction where very simple DNA transfer can happen. This can happen by one of two ways. I will just give you the names, okay? It's not mentioned in your syllabus. Just know one type is called conjugation, which is what I've shown here. Then you have another type called transformation. And then you have transduction. Okay? So here in this image, what is happening is this bacterium has its genomic DNA. Along with that, it has an additional DNA called plasmid. It makes an extra copy of its plasmid and it passes it down through this tube to this other cell. This cell didn't initially have that plasmid, now it has that plasmid. This plasmid is nothing but DNA, okay, a type of DNA. This has DNA that it did not initially have, so it has got new DNA, new genetic material variation. That itself is considered to be sexual reproduction here. Now, last part of Monera, let's speak of mycoplasma. Mycoplasma are also included in Monera because they do not have a nuclear membrane around their genetic material. But unlike all other bacteria, these mycoplasma do not have a cell wall. They completely lack a cell wall. Also, I told you that bacteria themselves are small. They're like super tiny. Among all of the bacteria, these mycoplasmas, wall-less organisms, they are the smallest of all. Okay, they are smaller than 1 micron also, some of them. Okay, so they're the smallest cells known, living cells known. They can survive with oxygen, but even in the absence of oxygen, they can still survive. That does not mean if you give it oxygen, it will die. With oxygen, they can survive, but even if oxygen is not there, they can still survive. And some of these species, mycoplasma species, are pathogenic. They are known to cause diseases in animals and plants. Okay. So one such disease that these organisms cause is a respiratory illness. Okay, something like pneumonia. That's why these organisms are also known as pneumonia like organisms. Okay, PPLO in short. You can see how they have a plasma membrane. There is DNA, there are ribosomes, but there is no cell wall. No cell wall, smallest known living organism. They can survive without oxygen. All of these are important points. So that is all we had to know about kingdom Monera. Now let's move on and learn about kingdom Protista. What do we already know about protists? Protists are organisms that are included under kingdom Protista. Okay. So we already know that they are eukaryotic because all prokaryotes were put under Monera. We were left with eukaryotes. So protist is one eukaryotic kingdom, Protista. Apart from that, we also know that they are unicellular, right? So all eukaryotes that were unicellular were grouped under this kingdom. So this kingdom includes organisms that are unicellular and eukaryotic. So when you compare the two kingdom system of classification which did not have Protista, in the two kingdom system of classification, organisms like Chlamydomonas and Chlorella, they were placed in plant kingdom. Organisms like Paramecium and Amoeba, which can move around, they were kept under plant kingdom. Now that we are considering organisms that are unicellular and eukaryotic to be included under Protista, these organisms that were earlier part of kingdom Plantae and kingdom Animalia are now present within kingdom Protista. So kingdom Protista has organisms that don't have a cell wall, it has organisms that have a cell wall, it has organisms that are non-photosynthetic, photosynthetic. So here the boundary of this kingdom is not very well defined. So it includes organisms that are similar to plants, similar to animals, it is similar to fungi. So it is kind of like a connecting link between kingdoms fungi, Plantae and Animalia. Okay. So within kingdom Protista, you find different kinds of organisms. Chrysophytes and dinoflagellates, both of these are photosynthetic. So we can say that they are plant-like. They are not in plant kingdom. They are similar to plants in a lot of features. Then we have euglenoids, which suffers from identity crisis. Like I mentioned earlier, this has some plant features and animal features also. Then we have slime molds, which are like fungi. And finally, we have protozoans, which are animal-like. We have to learn in detail about all of these organisms. Okay, so we'll start off. Wait, before that, we'll learn about some general characters of Protista. Mostly, they are organisms that are aquatic. Okay, they are mostly found in water habitats. Like I mentioned earlier, it forms a link with kingdoms Plantae, Animalia, and fungi. Some of these organisms have cilia or flagella for locomotion. Protists are slightly more advanced than Monerans because they are eukaryotic. Reproduction also is slightly more advanced. Here you can find various ways in which asexual reproduction happens. And whenever sexual reproduction happens, zygote formation is something that you can see very commonly. So sex cells or gametes form and they fuse. Zygote formation does happen every time during sexual reproduction. Let's speak of kingdom Protistas, chrysophytes first. Okay. Chrysophytes, include two different types of organisms. One is called diatoms and the other one is called desmids. They're also called golden algae or golden brown algae because of their color. These are some of the most beautiful organisms you can find on our planet. Okay. See, in Monera, I said there is blue-green algae or cyanobacteria. Here I'm saying there's golden brown algae. When we learn plant kingdom, I'll tell you there is green algae, red algae, brown algae. So algae are distributed across kingdoms, Monera, Protista and Plantae. Don't assume that the moment somebody says algae, it should belong to plant kingdom. Okay. So here these are also called golden brown algae because of its color. Okay. Now, where do you find them? You mostly find them in water, like I mentioned earlier. They could be found in fresh water or even in saltwater marine environments. These are definitely microscopic considering they are made up of a single cell. Obviously, a single cell cannot grow this big unless there are very rare cases, but they're mostly very, very small microscopic organisms. And they don't themselves have any locomotory structures. They are found floating on the surface of water. They are found in water. They can't be present deep inside the water because so deep inside the water sunlight cannot penetrate. They are photosynthetic, so they require sunlight. So they are found floating on the surface of water. As and how the waves in the water move, they will also move accordingly. It's not like they can decide I want to go right, left, northeast, northwest I want to take. They cannot decide that. We call that passive movement. Actively they are not spending energy to move to a particular direction. Along with water movement, they will also move. So that is called passive movement. They float passively in water and they are called planktons. Okay. So these are plant-like organisms that can photosynthesize. So we call them phytoplanktons. Like that we have zooplanktons also. Plankton is anything that floats freely on the surface of water. Okay. Now, most of them are photosynthetic. This is an NCERT trap. Here they are saying most of them are photosynthetic. Not all of them are photosynthetic. Some of the diatoms have lost the ability to photosynthesize. They do not have the pigment and the machinery required for photosynthesis. So they have stopped photosynthesizing. They can no longer photosynthesize, some of the species, but a vast majority of them are photosynthetic. This is important. Okay. Now, yeah, some of the amazing features about diatoms are about its cell wall. Okay. So normally when we say, when we speak of cell walls of plants or of fungi, you know how it's like a continuous structure. It's like a wall around the protoplasm, right? But here the cell walls are in the form of two halves, two shells, okay? And these two shells overlap on top of each other like a soapbox. That's so cool, isn't it? That's why they're called diatoms. Diatoma, I think in Greek means split into two or something. So the cell wall here is very unique. It is made up of two overlapping shells that fit on top of each other like a soap dabba. So here you can see this is a diatom's cell wall. Here this top one, this is one half black color and the red color bottom is the other half. The top one is always bigger, the bottom one is always smaller. The top one is called epitheca. Epi means above. The bottom one is called hypotheca. Hypo means below. Theca mostly is like a covering of sorts. Okay. So that is epitheca and hypotheca. That's just not the only unusual thing about the cell wall of diatoms. Mostly cell walls in most other organisms from bacteria to fungi to plants, it's made up of polysaccharides, right? It could be made up of chitin, lignin, cellulose, whatever, but generally there is a lot of polysaccharides present in the cell walls. Here, polysaccharides are present. It's not like they're completely absent, but they are present in very small quantities. That acts like a matrix, and within the matrix of those polysaccharides in the cell wall, silica is embedded. You know what silica is, right? Silica is SiO2. It is the same thing that is found in glass. If the same thing that is found in glass is found in the cell walls of these organisms, then the cell wall will appear like glass, doesn't it? Right? So here it is embedded with silica, which will give it a glass-like appearance. How cool, how cool. To look at them under the microscope, they're so pretty. And all of them have many different types of patterns, so it looks so beautiful under the microscope. And apparently jewelry designers take inspiration from diatoms for making jewelry. And also since they're so pretty, they're also called jewels of the sea. And like I mentioned, they literally look like glass. Look at this diatom. This is a micrograph of a diatom taken from under a microscope. Doesn't it literally look like a piece of glass? That is possible. It's appearing like that because the cell wall of these organisms are embedded with silica, the exact same thing that glass is made up of. Here, I'm going to show you one other image which looks so beautiful, so serene, so calming, so pretty. These are nothing but diatoms. Okay, so each organism that you look at here is a diatom. This is one diatom. It is appearing in this badami shape, whatever, oval, what do you call this? This shape. And there are some that are like triangular, there are some that are spherical, there are some that are spindle shaped, oblong. So you have many different types of shapes in which diatoms come. This is an arrangement. It looks like a glass ceiling on top of a church or something. But this is actually an arrangement, an artwork that is created by a diatomist called Klaus Klemp. What he basically does is he goes to different parts of his locality, wherever he goes. He collects water samples. He looks at the water samples under the microscope. He separates these diatoms based on their shape and size and then makes these beautiful arrangements and takes pictures of them from under the microscope. So it's very, very pretty. You can think about how much time and energy and focus and what else? Skill some creating art like this requires. So he's almost like very old right now. I don't know if he's still alive or not. You can check it on Google. His name is Klaus Klemp. Okay, so he is a diatomist who has made many, many such arrangements. So pretty to look at. They look like Deepavali diyas, very pretty. But all of them are microscopic. You can't really look at them and sort them. You have to look under the microscope the entire time while you're sorting them according to their size and shape. So this is crazy, crazy work. Yeah, look at how beautiful and glass-like they are. Now you might wonder, why is it that they had to have glass in their cell walls? Why couldn't they just be like normal other organisms having just cellulose and pectin and those sort of cell wall materials? See, again, there is a reason why anything exists in nature. The reason why cell walls of these organisms are made up of silica is because it offers them some benefit. What are some benefits that you can think of because of silica? I mentioned that they are like small glass pieces, right? Would you want to chew on glass? Would you want to eat glass? What will happen? Your mouth will get damaged. Your oral cavity will get damaged, right? Nobody eats glass. People could die if they eat glass, right? So same thing even for these organisms because there's so much glass like other organisms that feed on it, smaller fishes or maybe some small animals that feed on it, they will try to avoid it because they do not want to damage their mouth parts, right? So that way it offers them some defense. That is why it has been retained this way over so many thousands of years in the uh, during the course of evolution. Okay. Now, another important thing about these cell walls is while they are there, they are helping, protecting the diatom. But once the diatom dies, the contents inside the cell wall, which is called the frustule, this is called the frustule. The cell walls of diatoms is called the frustule. It cannot be destroyed because you know glass is non-biodegradable. No enzymes can break it down. Similar to how plastic, the toothbrush that you use today, if you leave it somewhere 500 years later, it's still going to be like that because microbes cannot act on it and decompose it. Very similarly, glass also cannot be decomposed. Of course, it can be recycled, but it cannot be broken down. So these organisms, when they die, the internal cytoplasmic contents, the protoplasm, that will get decomposed. But the outside shells will remain as such. Because the cell is no longer living, the shells will kind of sink to the bottom of the water body, and over millions and billions of years, many such diatoms have, given away their lives, they've lost their lives, they've died, and those frustules or cell walls have sunk to the bottom of the water body and formed a sediment which we call diatomaceous earth. Diatomaceous earth, I think I added a picture. Let me see. Yeah, it looks like this under the microscope. Okay. So this diatomaceous earth is beneficial to humans because of its porous and gritty nature. You see how the walls have many, many pores. You can't see this with your naked eye, of course. Under the microscope, tiny, tiny pores are visible. Because of those microscopic pores, these are used in filters where oils are being filtered, certain solutions are being filtered, syrups are being filtered for filtration purposes because they have naturally microscopic pores present. They are used similar to how we use filters for filtering our coffee, right? You do not want the milk cream to appear in your coffee, so you filter that because there is a pore size that can trap it. Here also very similarly, this pore size, since it's microscopic, it can even trap certain bacteria. Okay, so this can be used in filtration process. And I mentioned that these are like glass particles, very, very tiny microscopic glass particles that can be used for polishing. It can be used as an abrasive. Like if you want to wash certain utensils that have been charred, like you heated them for too long on your stove and it has been charred, it has like black color deposit on top of it, you want to get rid of it, what would you do? You would immediately rush and find Sabina, right? Sabina is that dishwash powder, very popular in the 90s, but right now you have Exo Vim and all of that. But Sabina used to have diatomaceous earth in it. Because of its small, very gritty nature, it acts like an abrasive and helps in removing all of the stains in your utensils very rapidly. Okay, and also it can be used for polishing because of its gritty nature. So diatomaceous earth is not a waste. It is very, very essential. It can be used for polishing, for filtration and all of that. Okay. Now, one cool feature I want to talk about diatoms, which is not there in your books, but I want to tell you anyway because it's cool. So I told you how it has two parts to its cell wall. The upper one is called the epitheca and the lower one is called hypotheca. Every time a diatom undergoes cell division, mitosis, like most other organisms, you know, the cell walls will get separated. If this parent cell had epitheca, hypotheca, they will get separated. This will become the epitheca of new diatom cell. Both of them will become the epitheca. That means the newer part of the cell wall that is forming will be the hypotheca. So if this forms the epitheca, this one, it will have the same size as this one. But if this red one becomes the epitheca, another smaller hypotheca is formed inside it. So this will be much smaller than the initial size. Every time cell division happens, one cell will have the normal size, the other cell will be smaller. Eventually, the cells of the diatoms will become smaller and smaller. Once it becomes too small, it will stop dividing asexually and it will form a sexual spore, and then the cell size is brought back to its original normal size. Okay, so that's something that is cool about these diatoms. So yeah, that's about diatoms. Like I mentioned, they are photosynthetic. Since they are photosynthetic, they are considered to be chief producers in the oceans. What do we mean by producers here? Producer here is not Karan Johar and all of those film producers. Producer here refers to a photosynthetic organism. See, there are many animals living in water bodies, right? They depend on plants for food directly or indirectly. But in water bodies, do you see big trees like mango tree, orange tree, apple tree that they can go pluck a fruit and eat? They can't, right? Because such huge plants you do not find in water bodies. In water bodies, most plants are like, or most photosynthetic organisms are like planktonic diatoms and dinoflagellates and tiny algae that float on the surface of water. They are the producers most. They are the photosynthetic organisms most. So that's why we say they are the chief producers. Chief producers here means they are very significant, very important kind of producers that you can find in an aquatic ecosystem. Okay, that's about our diatoms. Let's move on and learn about dinoflagellates. What does dinoflagellate mean? Dino here does not relate to dinosaurs. Dino, di means two. Flagellates obviously relates to flagella. So, these organisms have two flagella. Okay, most of them have two flagella. These are mostly marine and photosynthetic. Mostly again means that there are some that could be freshwater forms, right? So, they have different colored pigments. They could appear red, yellow, blue or green or even brown, depending upon what kind of pigments it carries. It's eukaryotic, it will have plastids. So within the plastids, pigments will be present. And here also the cell wall is kind of unique. Usually you see like a continuous layer of cell wall present around plant cells or fungal cells, but here cell wall is available or present in the form of plates, like armor. Like if you watch Mahabharata, you know how Bhishma and Duryodhana and Arjuna, all of these warriors, they have an armor, right, around them. Even if you watched Bahubali or whatever, you know that they have an armor and that'll have like different components, different parts. So this is also like that. That's why they're called armored dinoflagellate. So here the cell wall will come in different parts like this. Okay. So there will be furrows in between these plates. Okay, so these cellulose, it's made up of cellulose only, but it's not like a continuous layer. So it will have many furrows or groove sort of structures in between the plates. That is the characteristic feature of dinoflagellates. I told you that dinoflagellates, the name itself means it has two flagella, but again, not all of them have two flagella, some of them don't. So most of them have two flagella. How are these flagella arranged? They are arranged perpendicular to each other. There will be one longitudinal groove, like a vertical groove, and a horizontal groove. In the horizontal groove, one flagella will exist, that is called the transverse flagellum. See, this is horizontal, right? So the transverse flagellum will kind of arise from here, and it will be like this. It will go around and be like this. And the longitudinal one will be like this. Poink, like that. Okay, so that's how the two flagella are perpendicular to each other. Yeah. Similar to how blue-green algae are known to cause algal blooms, dinoflagellates also cause bloom. Okay. There's one characteristic type of uh, water bloom, algal bloom, or bloom in general that you can find because of dinoflagellates, and that is formed because of a dinoflagellate that is red in color called goniolax. Okay. So I told you how dinoflagellates can come in so many different colors, right? So some dinoflagellates that are red in color, they will occupy the entire surface of the water body, and the entire water will start appearing red like this, and that is called red tide. It might seem like, oh wow, water is usually so blue, now it is red color, so fancy and all of that, but there's nothing fancy about this. This also causes pollution in the water body. Many other organisms, depending on water for their survival, will be affected. These can, produce toxins and release toxins that can kill other animals and even humans if we consume them. So there's nothing beautiful or fancy about these. These are not good. Okay. So like I mentioned earlier, too much of anything is not good. It's bad. Yeah, some of these dinoflagellates have another amazing property, like a superpower. That is, it can emit light. Okay, the property where a living organism has the ability to emit light, it is called bioluminescence. Bio means living organisms, luminescence means light. So living organism producing light is called bioluminescence. Not all of them, some of these dinoflagellates have the ability to emit light. And why would they do that? Do they like to play light? Why are they emitting light? They emit light in order to distract its predator. These dinoflagellates are like small microscopic plants, microscopic photosynthetic organisms, right? There will be some other animals like amoebae or euglena that will want to feed on it. So if you know how you're trying to chase a lizard and kill a lizard, it'll let go of its tail and try and scare off the predator, right? Like that, this will emit light. So when this emits light, the predator could get scared and not attack it. So as a defense mechanism, it emits light. When it's not disturbed, it's chill to itself. But if you try to, you know, uh, stir up water, then it will start emitting light because it's scared. Okay, that's about dinoflagellates. Now let's move on and speak about euglenoids, one of the coolest type of protists that you can find. These do not fit into conventional boxes. Okay, so it has characteristics of its own. It's like a misfit. Like in your schools and other places, you will find rebels, right? Like the teacher will say you have to do this or this, and they'll be like, I don't want to do either. I want to do something else altogether. So these are like that. You can't put them in either boxes. So they're very different and unique that way. So these euglenoids are mixotrophs. One example for that is euglena. I think in your lower grades you would have learned euglena under protozoans, but right now you are being, you know, enlightened that euglena does not belong to protozoans. It's an entire different group called euglenoids. So here, these are mostly freshwater. Dinoflagellates are mostly marine. Euglenoids are mostly freshwater forms, and they're usually found in stagnant water. Stagnant water means still water. It's not flowing like a river water. It could be a pond or a lake. That's where it's mostly found. And yeah, speaking of the cell wall here, they do not have a cell wall. What they have instead is a protein-rich layer called the pellicle. Outside the plasma membrane, you can find a protein-rich layer which is called pellicle. Now, because they have pellicle here, the pellicle gives it some sort of flexibility. The pellicle, which is protein-rich, will provide some flexibility. Cell walls are rigid. They're mostly made up of polysaccharides and they're rigid. Having a protein that is kind of contractile, it gives it some flexibility to these euglenoids. Now, what did I tell you about euglenoids earlier? I told you they are a misfit. You can't group them either in kingdom Plantae or kingdom Animalia because it shares characters of both groups, right? So here you can see that these eukaryotic organisms will have plastids like chloroplast, which contains pigments similar to pigments that are found in higher plants. So it has the ability to photosynthesize. It knows a way to trap the sun's energy, perform photosynthesis, synthesize glucose, be self-sufficient. It knows it. But sometimes when the sunlight is not sufficiently available, it will have to engulf other organisms and eat. So when it has a flexible body shape, it allows it to engulf and feed on other organisms. Other plants, other photosynthetic organisms, they don't engulf other organisms when sunlight is not available. They don't have that mechanism. But these guys have that. And having a flexible body will ensure that they can kind of feed on other organisms when there is a need. Okay. And these also, whenever you've drawn euglena in your school, you may have usually drawn a single large flagellum like this. But euglena also has two flagella, one that extends out like this and one very small one that is retained within this region. Okay. Another amazing feature about euglena, though it is a very simple unicellular microbe, it is very, very clever. It has what is called photoreceptors, which are made up of proteins. Okay, similar to how we have photoreceptors in the retina of our eyes, rods, cones, and all of that. Like that, it has photoreceptors that can sense light. And then it has what is called stigma or eye spot. Okay, so with the help of the stigma or eye spot and the photoreceptor, which is very close to the flagella, they can detect the intensity of light, they can detect the direction of sunlight, and according to that, they will ask the flagella to take it to the region where sunlight is available so that they can perform photosynthesis. Obviously, since they have the machinery for photosynthesis, they will always try to prefer to synthesize their own food and eat. But God forbid there is no sunlight, then there's not much they can do. So they will end up becoming heterotrophic and feed on other organisms. Okay? Yeah. So I've mentioned this already. Now let's speak of slime molds. Slime molds are fungi-like. Why did I say that they are fungi-like? They are fungi-like because their mode of nutrition is saprotrophic. Okay, so they depend on dead and decaying organic matter for their nourishment. So where do you find them growing? They're saprotrophic. They depend on decaying plant and animal matter. So they are found growing on twigs, barks, fallen off leaves, and all of that. So when the conditions are favorable for their growth, I'll tell you what they do. These are also like small amoebae. Okay, so there will be very, very small cells. I'll draw it for you here. Okay, very, very small cells. Wrong choice of color. Very small cells like this. They are individual cells. Whenever the conditions are favorable, they release chemicals, chemical signals, and all of these over here, there is no cell wall. They will all come together, fuse to form a huge mass of huge cytoplasm with a lot of nuclei inside. Again, here no cell wall. This happens when the conditions are favorable. There is no cell wall. They're just spreading across like amoeba, across wherever there is organic matter present in the form of dead plants or animals. It will just start spreading out, secrete enzymes, absorb nourishment from there, divide, increase in size. Sometimes they spread across several feet in forests. You can see that. And let's say conditions become unfavorable. The temperature is not very optimum. There's not sufficient organic matter present around. Then what they do, they will start producing fruiting bodies. So this, when the conditions become unfavorable, will start producing fruiting bodies that kind of look similar to what I'm drawing right here, like this. And within the fruiting body, spores will be produced. When does this happen? This happens when the conditions turn unfavorable. So when these fruiting bodies burst open, they will release the spores, and the spores are carried by wind across to different places. So it's possible that some other place where the spore will land, conditions could be favorable. Right now where they're there, conditions are not favorable. So it's possible that wherever they land, conditions could be favorable. Over there, they will germinate and they will become plasmodia again, and then they will start growing. And these spores have cell wall. They have very thick cell walls. Every time, remember, whenever structures are being produced during unfavorable conditions, usually they will have very thick walls, because to resist the unfavorable conditions or to kind of protect the internal contents from the outside external harsh conditions, they will produce thick walls. I told you this when I spoke of spores being produced by bacteria also. So during unfavorable conditions, they will produce thick walls. So in the life cycle of these slime molds, there are phases where there is no cell wall, there are phases where there are cell walls, so you can't really put them into Plantae or Animalia. That was the issue that we had, right? So since these are eukaryotic and unicellular in one phase, you consider them as protists now, okay? Yeah. Now moving on to the last part of kingdom Protista, protozoa. Protozoa, as we know, are heterotrophic, they are animal-like. They could be predators or parasites. Predators means they kill other organisms and consume them. Kill and eat. Parasites means feed on or obtain nourishment from living organisms. Obtain nourishment from living organisms. That living organism is called a host. Okay. There are four major groups of protozoans that are classified based on their locomotory structure. Okay. What are they? Let's take a look. First, we'll speak of amoeboid protozoans. Example for that, very common, amoeba. These are found in aquatic habitats. You may find them in fresh water, sea water, or even on soil wherever there is moisture. I told you that this classification is based on how they move around, locomotory structures. Amoebae are known to move around using their false feet called pseudopodia. This is not new for you. Not only do they use pseudopodia for moving around for locomotion, they also use it for feeding, right? Yeah, capture their prey for feeding also, for locomotion also. This is shapeless organism. It does not always be like this. It will keep on changing its shape. So if there's some bacteria here and wants to feed on that bacteria, that will change its shape, engulf this, act like a friend, hug it, and then eat it off. Boom, gone. Okay, so they use the pseudopodia for that. And another important point in NCERT is, if the amoeba is a marine form, it lives in salt water, then some of them can have silica shells on their surface for protection. The moment you see silica, you should not think about diatoms. Okay, so even on amoeboid protozoans that are marine forms, you can find silica shell on the surface for protection. Some of them are parasitic, they cause infections. Example, entamoeba causes amoebic dysentery. Blue stools and all of that. Okay, so that's about amoeboid protozoans. Next, we have flagellated protozoans. Obviously, as the name itself indicates, they have flagella. Example for that is trypanosoma. They may be free-living or they also may be parasitic. Trypanosoma is known to cause sleeping sickness in humans. This is mostly seen in Africa. It is spread by a fly called tsetse fly. So what basically happens is it ends up messing up your circadian rhythm. Sleeping sickness means it's not like they cannot sleep at all. they will sleep at the wrong times. Okay, so there is a pineal gland present in your brain which is responsible for controlling your circadian rhythm, right? It'll let you know now it's day, broad daylight, it's bright outside, now it's time for you to wake up, it's dark now, you have to go to bed. All of those indications we get from our pineal gland. So this little guy goes up to your brain and messes it up so bad that you will feel sleepy during the day and you will feel restless at night. You can't go to bed at night, you can't sleep at night. So that is called sleeping sickness, and that's caused by this trypanosoma. I hate microbes that go and mess with your brain. I hate them. Yeah. So these are ciliated protozoans. This third type, as the name itself indicates, they are made up of cilia. Cilia is not only going to help in locomotion here, in organisms like paramecium, it also helps in feeding. In paramecium, you can see how the shape is like a slipper, shoe sole, right? It's called slipper animalcule also. So there will be this cavity present which is called the gullet. Throughout the surface of paramecium, you can find many tiny hair-like cilia present. They all keep beating and propel this organism forward. And when the cilia beats, okay, the food that is present outside also will be drawn inside so that this can feed on it. Okay, so that's about ciliated protozoans. What do we have next? Last one, sporozoans. Sporozoans do not have any locomotory structures. The reason they are called sporozoans is in their life cycle is because in their life cycle, if they have to complete their life cycle, there are some spore-like stages, and those spore-like stages are infectious. For example, if you think about the malarial parasite Plasmodium vivax or Plasmodium falciparum, you know that it will have sporozoite kind of a stage, which is a spore that causes infection. So that's why they're called sporozoans. We've mentioned Plasmodium twice in this lecture, once while I was talking about slime molds, once right now. Both the Plasmodia are entirely different. That Plasmodium in slime molds is a mass of cells, a complete combined cytoplasm with many nuclei, a multinucleate structure. Here Plasmodium is referring to a particular genus of protozoans. Okay, so it will have a spore-like stage in its life cycle. Plasmodium is its example. See, it does not have any locomotory structure. Next, we have to talk about kingdom fungi. Fungi are also pretty cool organisms. From what we already know, they are heterotrophic, which means they obviously can't prepare their own food. They depend on other organisms. And they are said to show a great diversity in morphology, their structure and habitat. That is where they are found usually. We say that fungi are cosmopolitan in distribution, which means they are found pretty much everywhere. They are found in the air around us, on the soil, in water, on top of organisms' body, inside organisms' body. They are pretty much found everywhere on our planet, which is what cosmopolitan means. They have a special preference, most fungi, for growing. They grow really well when the conditions are moist and the temperature is like not too high or not too low. It should be just warm enough and moisture. They grow there very well. You may have noticed how sometimes some food at home, like if you don't refrigerate it, it gets spoiled, right? So the reason why food spoils, the taste changes, the texture changes, the odor changes, all of that happens because some microbes would have started, breaking it down and producing certain acids and stuff. So that happens mostly during summers. The food that you would have prepared during the day, very early in the morning, tends to get spoiled by afternoon or a little early evening if you don't refrigerate it. But that rarely happens during winters. Even if you don't refrigerate it, it doesn't get spoiled very easily. That's because the temperature is cold and such temperatures do not favor the growth of fungi. Okay, so talking about its cell wall is made up of chitin, which is a type of polysaccharide. Okay, now how do we know about fungi? We know fungi from the food that gets spoiled, right? You may have seen these patches on top of bread if they're kept for too long in a kind of warm and dark place. They tend to grow there very well. You may have seen these kind of patches forming on fruits when they're not refrigerated or when they're not kept in an airtight or a chill, cold place. So fungi forms these kind of patches on moist bread and rotten fruits. Also, we eat fungi. Like, I don't eat fungi, but some of you all, of course, consume mushroom products, right? So, mushroom is a fruiting body that some type of fungi produce, and that is also a fungus. So, are toadstools. Toadstools are very similar to mushrooms in appearance, but they are usually brightly colored, and most of them are poisonous. You should not consume them. And they call toadstools because toads are usually found sitting and resting on top of these fruiting bodies. And then we also know fungi from yeast. Every time you watch like a baking video on YouTube, like all of these recipe tutorials and stuff, recipe videos, not tutorials. Tutorials is for dance, right? Yeah, recipe, all of those recipe videos. So, you know, they will add this activated yeast and then they will mix up the whole thing. So, yeast is used in the baking industry exclusively for making breads and cakes and stuff, which is why it is also called baker's yeast. And yeast is also used in the brewing industry for the production of alcoholic beverages like wine, whiskey, rum, brandy, and all of that. So they are also called brewer's yeast. So yeast is extensively used both in the baking as well as brewing industries. Now, we obtain many chemicals like many antibiotics from fungi. I told you about antibiotics when we discussed the benefits of bacteria as well, both bacteria and fungi. Like some bacteria and some fungi, they are known to produce chemicals that will limit the growth of bacteria, that will stop the spread of bacterial growth. So they are called antibiotics. One example for an antibiotic producing fungus is penicillium. The antibiotic that we obtain from it is called penicillin. Fungi are known to cause diseases in plants, similar to how most of the animal diseases are caused because of bacteria. In plants, most diseases are caused by fungi. You know the mustard leaf. There is a disease that affects mustard plants where on the leaf you can see white spots. This is called the white rust disease of mustard, and it is caused by a fungus called albugo. And wheat rust disease is another disease where you can see rust orange colored spots and stripes on top of the wheat stem. This is caused by a fungus called Puccinia. Okay. Next, most fungi are multicellular. However, there are few species that are unicellular. These unicellular fungi are called yeasts. Generally, the moment we say yeast, we will think about Saccharomyces cerevisiae that is used in the baking and brewing industry. That is also a yeast, that is also unicellular, but yeast does not mean there's only one species, Saccharomyces cerevisiae. There are many species of yeast, many genera of yeast. Candida is one yeast genus and Monascus is one yeast genus. So there are many species which are unicellular. Then if you're asking about why during Whitaker's five kingdom system of classification, we mentioned if it is unicellular and eukaryotic, it should belong to Protista. What business does it have being in fungi? This does not belong here. It has to belong to Protista. If you're thinking that way, then you know, in chemistry, you learn about so many different laws and so many laws have so many exceptions. Likewise, here we are talking about classification of 1.7 to 1.8 million species. It's kind of difficult sometimes to put them into 5 boxes only, considering sometimes that it shares characters between more than one organism. So the reason why yeast is put under fungi despite having just one cell, despite being unicellular, is because it is saprotrophic like fungi. It has some of them have cell wall material similar to fungi, and sometimes they form a mycelium-like structure similar to fungi, which is called a pseudomycelium. Since it shares some characters with the kingdom fungi or fungi in general, this is more suited here. So that rule is broken here, and this is like an exception. You can think of it like that. Okay. And most others are multicellular. Now, what is the body of fungi like? I told you that mushrooms are fungi and toadstools are fungi. Does that mean it is an organ? What level of organization can we see here? So in fungi, the organization is very simple. There's no organ level of organization. There are just loosely, loose tissue sort of organization that you can see here. Basically, if you look at the body of a fungus, for example, here is a spoiled tomato. It is kind of rotting. There is fungus growing on top of it. You take this and look at it. It kind of looks like this if you magnify it a little bit. If you further magnify it under the microscope, here's what it looks like. You can see small thread-like structures present. Okay. So these small thread-like structures are called hyphae. So these are hyphae, very small thread-like structures. This is hyphae. And when the thread-like structures form a network of sorts, then you call it the mycelium. Okay, so the network of hyphae is called mycelium. The thread-like structures is called hyphae. I told you how morphologically there is diversity in fungi. The structure and form differs from fungi to fungi, from species to species. So basically, one major difference that you can find in mycelia of fungi is that some of them have cross walls present between cells. I told you they are filamentous, right? I mentioned that they are long, slender, thread-like structures, which means cells are attached end to end, and within this thread, there are cells that are attached end to end like this. This is called filamentous nature. So in some species of fungi, you can see that between two cells, there is a very distinct septa or a cross wall present, such that each cell is made up of only one nucleus. If a fungal hypha or mycelium has these sort of septa or cross walls, you call such a mycelium septate. So here is an actual septate mycelium. Under the microscope, you can see how the cross walls are very clearly visible here, right? You can see them clearly. This is kind of like a more advanced type of mycelium. In some primitive fungi, you don't find septate. If you don't find septa or cross walls, the mycelium is said to be aseptate or cenocytic. Aseptate means septum is absent. Here you can see there is like a continuous tube where multiple nuclei are present. Okay, so continuous tubes filled with multinucleated cytoplasm. This is an important term to remember. We call it cenocytic. Okay, now speaking of their mode of nutrition, they show a little bit of variety in that as well. Most of them are saprotrophic, which means they obtain their nourishment from dead and decaying organic matter. Few of them cause diseases and act like parasites, in which case they are dependent on a living host for their nourishment. Some of them are symbiotic, which means they are in a mutual relationship with another organism where because of that interaction, both of them are getting benefited. Okay, so for example, you have lichens. Lichens is where fungi and algae both are interacting with each other. They are living together as a unit. Fungus, having filamentous nature, helps algae in absorption and also provides shelter. In return, alga provides fungus with food. There is another association called mycorrhiza. Myco means fungus, rhiza means root. Okay. So this association is between fungi and roots of higher plants, where again fungi helps in absorption and plants provide the fungus with food. It's a mutually beneficial relationship. It's not like the fungus is taking advantage of the algae or the plant. They both benefiting out of that relationship. Okay. So yeah, that's about the modes of nutrition. Speaking of reproduction, in fungi, you can find vegetative reproduction, you can find asexual reproduction and sexual reproduction. I'm sorry for those of you who had your earphones on. I apologize from the bottommost part of my heart. I didn't mean to cough in your ear like that, but sorry guys. So here you can see vegetative, asexual as well as sexual reproduction. We call it vegetative reproduction when no specialized structures are formed. Whatever hyphal cells are there, whatever vegetative cells are there, these cells are there, they themselves will get involved in the reproduction process. Then you call it vegetative reproduction. Okay. What is asexual reproduction? In both asexual reproduction and sexual reproduction, different types of spores get produced. If spores are getting produced as a result of mitosis, then they are called asexual spores. So, you also call them mitospores. If spores are being formed as a result of meiosis, then you call them myospores, and those are sexual spores. In asexual reproduction and sexual reproduction, in both cases, spores are produced. But during the production of spores, if mitosis is happening, then it is an asexual spore. If meiosis is happening, then it's a sexual spore. Okay. More about the different types of vegetative, asexual and sexual reproduction we will analyze now. First, we'll talk about vegetative reproduction. It is of three types. There is fission, budding, fragmentation. Fission and budding, both of that can be seen in unicellular fungi such as yeasts. You know what fission is? I already told you when I told you about binary fission in bacteria. It's very similar. What happens here? So you have one small cell. That small cell replicates its DNA, sends it to opposite poles, and then it becomes kind of elongated like that. It grows, replicates its DNA, and then it will divide. From one cell you get two cells. That is called fission. What happens in budding? In budding, what happens is you have one cell. This cell will produce a small projection of sorts. If you look here in fission, both the cells that are formed are of equal size, but here that is not the case. The bud will be very, very small. It's possible that one cell can have many buds attached to its surface of small size. Eventually, this bud attached to the parent cell can produce its own smaller bud. So this way, smaller projections will keep arising. DNA replication will happen. That DNA is sent to the bud. Eventually, if this bud detaches from the parent cell, that is equivalent to a new organism. That is how budding helps in reproduction vegetatively. Here you don't see any specialized structure forming. There is no sporangium or fruiting body or none of that is forming. The existing vegetative cells are themselves dividing and giving rise to new cells. So it is called asexual reproduction. Let's see what happens in fragmentation. I told you already that hyphae are long chains of cells. So let's say by mistake or God forbid, by mistake, somebody decided to walk over this fungus and this cell got damaged in between. And this one long chain, which was part of one organism, has now broken into two parts. It has become two fragments. Fragments means what? Pieces. Okay. So now that this has become two pieces, each one will behave like a new organism. From one you got two, that is also equivalent to reproduction, right? So here again, no specialized structures are forming. Direct vegetative cells themselves are taking part and giving rise to new organisms. So you call this vegetative reproduction. Now, speaking of asexual reproduction, you have three different types of spores that can be found. First type of spore is called conidia. They are also referred to as conidiospores. I'll tell you what happens here. Let's say this is like some log, a log of wood within which fungi are growing. Okay, so that is the substratum. It could be soil, it could be anything. For now, let's assume there is fungi growing there and its hyphae, you can't see this color, I'll use yellow color. So its hyphae are kind of spread across here. There's sufficient food available, there is sufficient space available. So they want to spread out. They want to increase in number. So when they want to increase in number, one of the ways in which they can do that is by producing spores. Asexual spores is easier. Okay, so they don't have to look for an opposite mating type hyphae or anything. With the existing hyphae only, asexual reproduction can happen. Now that this is inside the substratum, once there is sufficient nutrients and everything, what happens is, these hyphae will start producing upright branches like this. Okay, so these upright branches are called aerial hyphae. Initially, they were present near the surface or just above the surface, you can say. That is called subsurface hyphae. Once there is sufficient nutrition, they will want to increase their number, because ultimately the biological purpose of all living organisms is to make sure that they survive well, they continue to reproduce, and their generation, their species continues. So these guys also want to do the same thing. They want to ensure that more of their own kind is produced when there is sufficient nutrition available for them. So they are going to produce hyphae, upright, that are called aerial hyphae, and at the tip of this aerial hyphae there will be branches that are called conidiophores. Here I said conidiospore. Now I'm saying conidiophore. Conidiophore is not F-O-U-R-4. It's not F-O-R-E, forearm 4. It is P-H-O-R-E-4. P-H-O-R-E-4 means something that bears something. Okay. So here we are talking about conidiophore. That means this is a stalk sort of thing that bears conidia. If we say antheridiophore, that means that stalk kind of bears antheridia. If you call it archegoniophore, that means the stalk bears archegonia. So here we are calling it conidiophore, and this stalk, at the tip of the stalk, conidia will start forming. So conidia are these small spherical spores that are produced at the tip of the conidiophore. They will get pinched off one by one, just like you remove and pinch out chapati dough for making rotis and fulkas, right? So that way it gets pinched off, and these are called conidia or conidiospores. Eventually, when they reach the right substratum, they will germinate and give rise to new hyphae. That is what conidiophores are. Now, every time conidiospores are. Now, every time we're learning about these different types of spores, there's two things that we should keep in mind. Firstly, is it flagellated? Can it move around? Is it motile or non-motile? Secondly, is it produced inside a specialized structure or is it produced outside a specialized structure? Some specialized structure will exist, right? It could be a fruiting body, it could be anything. If there is a specialized structure and it is produced outside that structure, then you call it exogenous. It's generated outside. Exo means outside, genus means generated. So it's generated outside, you call it exogenous. Here you can see how the conidiophore is a special structure at the tip of which these conidia are present. They are present outside. It's not like there's a closed structure within which the spores are produced. So you call this exogenous. Exo meaning outside. Non-motile because obviously you can't see any flagella, any locomotory structure here. They cannot move by themselves. They are dispersed by wind currents. Okay. Yeah. Next you have sporangiospores. Sporangiospores, as the name itself indicates, are structures that are produced within, they are spores that are produced within structures called sporangia. Over here you can see a special sort of a structure which I am drawing. This is an enclosed structure. This is the sporangium. The spores are produced inside the sporangium. So they are called endogenous spores. Endo means inside. These spores again do not have any locomotory structure, so they are non-motile. Now, sporangiospores are those spores that are produced inside sporangia. Okay. They may be motile or they may be non-motile. If they are motile, they are called zoospores. They will have flagella. If they are non-motile, you call them aplanospores. Aplanospores are also sporangiospores. The next zoospores are also sporangiospores. But the difference is aplanospores are produced by land fungi. Zoospores are mostly produced by aquatic fungi because only when there is water, flagellum can be used to swim around, right? One matrix, liquid matrix has to be present for flagella to function, right? Now, these are zoospores. As you can see clearly, they are produced inside a sporangium called zoosporangium. They have flagella, right? So they are motile. They can move around. They are produced inside the zoosporangium, so they are endogenous. That's about the three different types of asexual spores. Now let's learn about the sexual cycle in fungi. There are three important steps that takes place in the sexual cycle of a fungus. What are they? Whenever sexual reproduction happens in fungi, similar to how in higher animals and plants you have a male parent and a female parent coming together, you don't have male and female gender or sorts here in fungi. You have what is called opposite mating types. Okay, they will belong to the same species. They will morphologically more or less look similar, but they are different with respect to the kind of genes that they carry. If they carry some genes, then they are considered to be plus mating type. If they carry some genes, some other genes, then they are called minus mating type. Okay, so or you can call them strains also. You don't call them male, female here. So here you have a let's say they're growing here. This is a substratum. Same species, two mating types fungi are growing. First thing is, if they belong to the same species and they are of opposite mating types, one is plus, one is minus, then between them sexual reproduction can happen. Another important thing that we must keep in mind about fungi is that unlike most plants and higher animals, these are not diploid. The normal hyphae that you come across in fungi, they are naturally haploid. In all species, vegetative hyphae is haploid. In us, we are diploid. Most plants are diploid, right? Most plants I mentioned, not all of them. Most plants are diploid. More about which ones are diploid, which ones are not, we'll learn in the next chapter. For now, just remember, in a majority of fungi, the vegetative filament hyphae, that is haploid. You have two haploid hyphae of opposite mating types belonging to the same species coming, across each other. Okay, so if they're coming in close proximity, it's not like they're walking towards each other or anything. If they come across each other while they're growing, then what happens is obviously there will be some chemical communication, and after that, one cell from this, one cell from this will fuse. Some fungi produce specialized sexual structures, some fungi produce specialized sexual cells, some of them will not produce anything special, the vegetative cells themselves will fuse. So it depends from species to species. For now, just understand one cell from the each of the two mating types will come close to each other and they will end up fusing. So this first step is called plasmogamy. Okay. So when fusion happens, the protoplasm will fuse, but the nuclei don't fuse. The haploid nuclei will stay separate, distinct within the same cytoplasm. So here you have two haploid nuclei present. Since there are two haploid nuclei, this structure is called a dikaryon. That cell can be called a dikaryotic cell, and that phase is called a dikaryophase. As a consequence of plasmogamy, the result is dikaryophase. It varies from species to species for how long the dikaryophase will last. In some of them, immediately after the plasmogamy has happened, the nuclei also will fuse. But in a lot of fungi, it remains in the dikaryophase for a long period of time. This dikaryon cell itself will continue to divide to give rise to many more dikaryon cells. So that way, it will occupy a large period of time. Okay, so it will go on for a long period of time. So then what happens eventually is the second step takes place. That is called karyogamy. Karyon means nucleus, gamy means marriage. So karyogamy, I wrote it as karyongamy. Karyogamy means the nuclei will fuse. When two haploid nuclei fuses, what forms? The zygote forms. As a result of plasmogamy, dikaryon formed. As a result of karyogamy, the zygote will form. The zygote will undergo the third step, meiosis, following which haploid spores will be produced. When these spores germinate, you get new hyphae again. Since these spores are produced as a result of meiosis, they are considered to be sexual spores. So typically in the life cycle of a fungus, you can come across three distinct steps. First, plasmogamy, following which a dikaryophase will form. That can vary from species to species how long a fungus stays in the dikaryophase. Following that, sooner or later, karyogamy happens, resulting in the formation of a zygote, which is diploid. The zygote undergoes cell division meiotically to give rise to haploid spores, which when they undergo germination will give rise to new hyphae. Okay. So that's the whole story of how sexual reproduction happens in fungi. So the three steps are plasmogamy, karyogamy, and meiosis in that exact same order. You can't expect meiosis to happen in haploid cells. So for meiosis to happen, the cells should have become diploid, right? So plasmogamy, karyogamy, after that only meiosis can happen. Next. Speaking of further classification of fungi, I told you when we were discussing the criteria that Whitaker used for his classification system, how reproduction can be used as a feature or a criterion for classifying. We'll learn about that now. See, when we spoke of R.H. Whittaker's classification, we had used uh, cell structure, body organization, mode of nutrition, all of that. Here we will use reproduction, and also we will use the morphology of the mycelium as criteria for classification. Also, what kind of fruiting body it will produce, that will be used as a criterion. Firstly, morphology of the mycelium. Is it septate? Is it aseptate? That is one thing. If it is aseptate, it is a primitive feature. If it is septate, it is more like an advanced feature. All the aseptate mycelia containing fungi, they are grouped under what is called phycomycetes. Phycomycetes is also called algal fungi. Some of them are aquatic like algae, and some of them have walls that are made up of cellulose, which is why they are called algal fungi or phycomycetes. Phyco means algae. Phyco, the term phyco relates to algae. Phycology is the study of algae. Okay. Then what are we left with? Mycelia that have cross walls or septa. For further classifying this, we will look at how do they undergo sexual reproduction, what kind of fruiting bodies do they produce. So here we have three types: ascomycetes, deuteromycetes and basidiomycetes. Ascomycetes are also called sac fungi. I'll tell you why they are called so. Basidiomycetes are also called club fungi. Deuteromycetes are called imperfect fungi. Poor things. Okay, so let's start off with phycomycetes. Example for phycomycetes include Mucor, Rhizopus and Albugo. Rhizopus is the bread mold that I showed you earlier. Albugo, I told you that it produces those white spots on mustard leaves. It's the same fungus. Both of these along with Mucor belong to phycomycetes. Where do you find them? Like I mentioned earlier, they're mostly aquatic. I told you that this causes disease in plants, so some of them can be obligate parasites on plants. How is the mycelium here? We know it is primitive type, it is aseptate. If it is aseptate, it's obvious that it will be cenocytic. How do they undergo asexual reproduction? Since most of them are aquatic, here zoospores will get produced. If they are terrestrial species, then aplanospores also could get produced. How do they undergo sexual reproduction? So here you can see specialized cells will form. I'll show you here, wait. So you have two different hyphae of two opposite mating types. I've represented one in pink and one in white. So let's say this is plus, this is minus. They will produce specialized structures. They will come close to each other, fuse. First plasmogamy will happen, then karyogamy will happen and the zygote will form. This is the zygote, you can see this. Over here, plasmogamy has happened. Here, karyogamy has happened. As a result of meiosis, you can see how the diploid zygote is formed. The zygote itself develops a thick wall around itself and becomes a spore called the zygospore. This zygospore will undergo meiotic cell division to give rise to spores, which when they germinate will give rise to new hyphae. Okay. So for the formation of gametes here, sometimes both the gametes will look similar, sometimes they look dissimilar, sometimes they have a specific dissimilarity. So when they're very similar to each other with respect to their morphology and size, they are said to undergo sexual reproduction by isogamy. Iso means similar. Anisogamy means one gamete will be larger compared to the other. Oogamy means specifically the female will be large and non-motile and specifically the male will be small and motile. That is called oogamy. So in one of these ways, the fusion of gametes can take place. Okay. So zygospore is the sexual spore that is formed here. Reproduction can happen by isogamy, anisogamy or oogamy. Now, Let's speak about the second type of fungi, the second group of fungi called ascomycetes. They're called sac fungi I mentioned, right? These are multicellular. Most of them are multicellular. However, the unicellular yeasts belong to ascomycetes. These are saprophytic like most fungi, decomposers like most fungi, can be parasitic too like some fungi, and this is important, they are found to grow on dung. We call that coprophilous. The mycelium and ascobasidiodeutromycetes, all of them are branched and septate. Okay. Here, asexual reproduction happens by conidia. I will not go ahead and explain what conidia is one more time. I just told you what that is. How does sexual reproduction happen? I'll tell you how. So here you have fungi of two opposite mating types. Let's say this is plus, this is minus. Opposite mating types, they produce specialized reproductive structures that are called gametangia. Okay. Then we'll not get into the details of what they are called. First, karyogamy will happen, as a result of which you can see the dikaryophase. In both ascomycetes and basidiomycetes, the dikaryophase will last for a long time. This dikaryon cell that you can see over here, okay, okay. So this is one cell that has two nuclei, common cytoplasm. This will undergo cell division and give rise to many more dikaryon cells. Eventually, these dikaryon cells will produce sac-like structures that are called asci. Singular is ascus, plural is asci. Once this is formed, these cells will undergo karyogamy. As a result of karyogamy, a diploid zygote will form. The zygote will undergo meiosis and give rise to 4 spores. From each meiotic division, you get 4 spores, right? It will produce 4 spores. Sometimes these 4 spores undergo another round of mitosis to produce 8 spores. And these spores are called ascospores. Around the ascospores, the remaining part of the hyphae, which did not fuse, where karyogamy or plasmogamy did not happen, those vegetative cells will around it produce a fruiting body that is called the ascocarp. Carp means fruit. This is the ascocarp. Within the ascocarp, you have sac-like structures that are called asci. Within the asci, the zygote will undergo meiotic division to give rise to spores that are called ascospores. Once the ascospores are liberated, they will germinate to give rise to newer hyphae. Okay, so that's about ascomycetes. Here's an image that will show you what an ascocarp looks like, what an ascus looks like under the microscope. You can see this is a picture. Within this, you can find 1, 2, 3, 4, 5, 6, 7, 8 ascospores. Ascospores number will always be either 4 or 8. It is not like there can be 5 sometimes, there can be 7 sometimes. That's not what it is. If immediately after meiosis 4 spores are produced, they can be released, or after those spores are produced, each one of them will undergo mitosis and give rise to 4 more spores, and in each ascus 8 spores will be there. Okay, linearly they will be arranged like this. This is the ascus. These are the ascospores. Where will these ascospores be located? They are located inside this fruiting body that is called the ascocarp. If this is the fruiting body, you can see the sacs here, can't you? Can you see this? Within these sacs, you can find the ascospores. So the sexual spores in ascomycetes is ascospores. Ascospores are located inside sac-like structures called asci. That is why it's called sac fungi. And where are these sacs present? Inside the fruiting body is called Ascocarps. How are the ascocarps produced? They are produced by the remaining vegetative cells in the hyphae. After plasmogamy, karyogamy, remaining cells that are not involved in plasmogamy or karyogamy will be there, no? They will produce this, okay, the vegetative cells. Next. Yeah. Similar to how mushrooms are eaten, mushrooms are basidiomycetes. There are some species of ascomycetes from which fruiting bodies are, collected and consumed as a delicacy. You can find mostly in Northeast India. People consume this. They are called morels and truffles. They are available for consumption. Apparently, they're very expensive. So, some other examples for ascomycetes are Aspergillus, claviceps, and Neurospora. Just like how Drosophila is used a lot for genetics experiment, Arabidopsis plant is used, and monkeys and rabbits and hamsters are used for many drug testing and all of that. Neurospora fungus is also used a lot for research in genetics and chemical research extensively in biochemical and genetic work. Okay, so that is Neurospora. Remember this. Next, we'll talk about basidiomycetes. Basidiomycetes are commonly known as club fungi. It includes mushrooms, bracket fungi, and puffballs. You may have seen these kind of ear-like fungi growing on top of logs. They're called bracket fungi because they also look like brackets. And these are called puffballs. All of them belong to basidiomycetes. These are found to grow, as you can see here. Mushrooms, where do you find them? You find them in the soil, right? You find these bracket mushrooms on top of tree logs, on tree stumps, and it can grow on living plant bodies as parasites as well. They cause diseases such as rusts and smuts. The mycelium here again is branched and septate. Only in phycomycetes it's unbranched and cenocytic. I'm sorry, it's aseptate and cenocytic. In every other group, it is septate and branched. Yeah, so in this group, you don't find asexual spores produced, but fragmentation happens very commonly, and that is how they reproduce vegetatively. They do not have specialized sexual organs, specialized sexual structures or cells. The vegetative cells themselves will take part in sexual reproduction here. What happens during reproduction here is very similar to what happened during reproduction in ascomycetes. You have two opposite mating types. They come together close to each other. They're both haploid, 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 haploid. And then when karyogamy happens, dikaryophase is what you can see. Okay, so here you have haploid plus haploid. Eventually, did I say karyogamy? Eventually, karyogamy will take place. Once karyogamy takes place, the zygote is formed. The zygote here will form inside structures, club-shaped structures that are called basidia, basidiomycetes, basidia. The vegetative cells will give rise to a fruiting body that is called basidiocarp. Remember, fruiting body, carp. Carp means fruit. Basidiocarp. Over here, on the surface of basidia, spores are produced. 4 of them. They are called basidiospores. Okay. So these basidiospores are exogenous because they are being produced at the surface of the basidia. But we saw how ascospores are produced inside the sac. So these are endogenous. They are endogenous, and basidiospores are exogenous. Once the spores germinate, they will give rise to new mycelium. So here is an illustration of the same. You can go through this in your notes. So yeah, the same thing is described here. Some common examples of basidiomycetes are agaricus, which is the common mushroom, Ustilago, which causes smut disease in corn, Puccinia, which causes rust disease in wheat. About this I had already told you in the beginning of the class, like at the beginning of fungi. Yeah, now let's talk about deuteromycetes. They're commonly known as imperfect fungi. I'll tell you why. See, as living organisms, as a biological species, we all have a purpose, that is to make sure that even after we are gone, our species continues to survive. And given sexual reproduction and asexual reproduction, sexual reproduction is always considered advantageous because it brings about variation in a population. And as a result of that, populations can adapt better to changing environments and the chances of a species survival will increase, right? So here, sexual reproduction is considered as the perfect stage of reproduction or the perfect cycle. Asexual is considered like imperfect, you can say. In deuteromycete fungi or fungi that are grouped under deuteromycetes, one of two things have happened. Either we do not know how they reproduce sexually. Do they reproduce by producing ascospores or basidiospores? We don't know about that. We haven't yet discovered how they reproduce asexually in those species. Since we don't know about it, we've grouped them under deuteromycetes, assuming that they only undergo reproduction by asexual means because we haven't yet discovered it. Or it is also possible that the same fungus, one fungus has been given two different names, not knowing that it's the same thing. And one has been grouped in deuteromycetes, thinking we don't know its sexual cycle, thinking its sexual cycle is absent there. We've grouped that in deuteromycetes, and the sexual cycle of this fungus could be included as an entirely different species in deuteromycetes or ascomycetes. Okay, so that also is possible. So as and when they discover how sexual reproduction happens, as and when they bring about connections between these different groups of fungi that are kind of dispersed across different groups, they will either move it to ascomycetes or basidiomycetes. If they ever figure out that, okay, this thing that we're looking at here is the same thing as that one, we assume that it did not undergo sexual reproduction, but look, it's the same thing. So then if they see that it reproduces by producing ascospores sexually, then they will move it to ascomycetes. If it's reproducing by producing basidiospores, then you move that to basidiomycetes. Okay, so eventually they get moved out of deuteromycetes once those connections and links have been established. Okay, so that's about deuteromycetes. We call them imperfect because we don't really know yet whether they reproduce sexually or they don't at all, or we have not kind of made those connections. Okay, yeah. These deuteromycetes reproduce only by asexual spores called conidia. Mycelium is septate and branched. They, like most other fungi, are decomposers and help in cycling of nutrients and minerals. Examples, you have three: Alternaria, Coletotrichum and Trichoderma. Okay. Trichoderma polysporum, the same fungus that is used in production of immunosuppressive drug, cyclosporin A, that belongs to deuteromycete. All right. Yeah. So here is a summary table for you. Here you can see the different types of spores that are produced: sexual spore, asexual spore, what kind of fruiting body, what is it called, and all of that. Okay. And also to help you memorize all of the examples, I've added all of the examples over here in one slide for your reference. You may revise all the examples from here. So with that, we've come to the end of Kingdom Fungi. About Kingdom Plantae and Kingdom Animalia, we learn very briefly in this chapter because there's a whole big chapter for Plant Kingdom and Animal Kingdom separately. So what are organisms belonging to Plant Kingdom? They're definitely eukaryotic. They contain chlorophyll, which is why they are capable of performing photosynthesis. Most of them are autotrophic. However, some of them are partial heterotrophs. Which ones are those? They are insectivorous plants like the Venus flytrap, like this is a Venus flytrap is an insectivorous plant. Why are we calling them partially heterotrophic? They have chlorophyll, they are green plants, they can perform photosynthesis. Why do they trap insects in that case? The reason why they trap insects is because in the kind of habitats that these insectivorous plants grow, a major nutrient, a macronutrient that plants require to grow, which is nitrogen, is not sufficiently available in the soil. Without nitrogen, plants cannot grow, or even we cannot grow, because like I told you earlier, nitrogen is a very important macronutrient. We need it to build muscle and produce protein. If a cell has to divide and replicate its DNA, nitrogen is essential. So it's needed for normal growth and development in somewhat large quantities only. So if they are not able to obtain nitrogen from the soil, then they will trap insects. They will have one or the other mechanism to trap insects, like they will have a pitcher which is slippery, and if an insect comes and lands there, it will slip down. Or they will have like a trap kind of a mechanism which can trap these insects, and eventually they will secrete certain enzymes that will break it down. And from that nitrogen, they will absorb and utilize it for their growth and development. So they're not like entirely dependent on other organisms for survival. They're not complete heterotrophs. Neither are they completely self-sufficient and independent. So we call them partial heterotrophs. Some of these plants are parasitic, which is very, very rare. Like for example, this is a plant called Cuscuta. This does not perform photosynthesis. This will directly penetrate its roots, specialized roots called hostoria, into the host plant's phloem where food is getting transported, and directly take ready-made prepared phloem food from there. So that is parasitic. This is Venus flytrap, and this is bladderwort. Cuscuta is there. Okay. Yeah. Plant cells have plastids like chloroplasts. They do have a cell wall that is mostly cellulosic in nature. It includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms, about which we will learn in detail in the next chapter. There's something called alternation of generation that you can see in plants. All plants, every plant from algae to angiosperms. More about that we learn in the chapter Plant Kingdom. Yeah. So this is not Plant Kingdom, this is Kingdom Animalia. Okay. So these are multicellular heterotrophic eukaryotic organisms. They do not have cell walls. They are heterotrophic, means obviously they cannot synthesize food by themselves. They depend on plants either directly or indirectly for food. They are ingestive heterotrophs. So inside their body, there is an internal cavity where food is consumed and it gets digested there, broken down there. They store reserve food in the form of glycogen or fat. Like I mentioned earlier, they are holozoic in nutrition. They obtain nourishment by ingesting food, taking food into the body. They show a definite growth pattern. Once they grow into adults, adults will have like a specific shape and size in most cases. In advanced, higher animals like us, there is elaborate sensory and neurotransmission mechanism or neuromotor mechanism. Most of these animals are capable of locomotion, again most, because poriferans can't move around, barnacles can't move around, right? So most of them are capable of reproduction. They undergo sexual reproduction by what is called copulation or mating, after which there is a brief period during which embryological development happens, and eventually the baby develops or the young one develops. Okay, more about Kingdom Animalia you will learn in zoology. And now we've reached the last part of the chapter. We've done with all of the 5 kingdoms. There are some organisms which you can't even call organisms really. So we'll talk about some of them right now, starting with viruses. You may have learned in your lower grades how viruses they have some features like living organisms and a lot of features like non-living things. So you can't really put them into either of the 5 kingdoms. So viruses are basically non-cellular, meaning they do not have like a proper cellular organization like most other organisms do. There is no cell wall, cell membrane, there's no cytoplasm, there's no ribosomes of their own. They don't have a lot of enzymes like other cells. They do have a few enzymes that are required for their infection and replication and stuff. But they don't have a cellular structure like other organisms, which is why they are called non-cellular. And they are obligate parasites. Unless they are inside the host cell, they are equal to dead. They are inert like non-living things, and they are crystalline in nature. Once they infect the host cell, they will take over the cellular machinery of the host cell. They themselves do not have ribosomes. They themselves do not have enzymes required for replication or protein synthesis or whatever. And they will enter the host cell and they will make use of the host's ribosomes, host's enzymes, and you convert that into a viral factory and make more copies of the copies of themselves and they will end up destroying the entire host cell and get out and infect more and more cells. Virus means venom or poisonous fluid. They are rightly called so. Yeah. Talking about how we got to know about the existence of viruses on our planet, there are 3 scientists' contributions that you must learn. The first one is Dmitry Ivanovsky. In the year 1892, he was studying a disease found in tobacco plants where the leaves were affected. On the leaves, you could find like a mosaic-like pattern. There was yellowing and patches forming, and they called that the tobacco mosaic disease. Back then, the only thing, the small microbe that they knew of that could cause these infections was bacteria. Okay, they didn't know that something smaller than bacteria could exist. So he expected that it could be a bacterial infection. He wanted to try to take out or extract the bacteria from this plant and observe that. So he kind of made an extract of this plant and he tried to filter that using bacteria proof filters. What are bacteria proof filters here? See, you have filters, meaning any filter will have pores, right? So if you want to sieve some flour or something, then you will use a filter that has small pores. If you want to filter milk, you will use a different kind of filter. So like that, you have different filters of different pore sizes. In microbiology, you have filters which have like extremely small pore size, so small that bacteria cannot pass through it. They are called bacteria proof filters. If you pass a fluid or a liquid or anything through it, if that fluid has any bacteria, it will get trapped in that filter, it will not be able to pass through it. The pore size will be so small. He expected that it could be a bacteria and he passed it through a bacteria proof filter, but turns out even after passing it through the bacteria proof filter, the virus could still pass through it. The infectious agent could still pass through that filter, which meant that the size of this particular infectious agent is much, much smaller than bacteria. So he figured that out. Okay, so that was his contribution. Later, M.W. Bejanik in the year 1898, what he did was he also continued to study the same tobacco plant. He took an infected tobacco plant and a healthy tobacco plant. Okay, so this one, let's say, is infected. This one is healthy. The infected one obviously has a lot of mosaic pattern and patchy yellow color. He took out this plant's leaf and its extract. Okay. And introduced that into the healthy plant. This was healthy. But after he injected this infected plant's extract to the healthy plant also developed infection, and the leaves of the healthy plant also started showing a mosaic pattern. Later, he called this fluid, the extracted fluid, as contagium vivum fluidum, which means infectious fluid. And it is M.W. Bejanik who coined the term virus for this new pathogen. Then you have W.M. Stanley, who for the first time crystallized the virus, and after crystallizing the virus, he analyzed the crystal and he figured that the crystals were made mostly of proteins. That was his contribution in the year 1935. Now, what else do we know about viruses? I already mentioned they are inert outside the host cell. Unless the host cell is there available for them, they will not be able to carry out any metabolic reactions. They require absolutely the host cell's machinery to make more copies of them and reproduce. By themselves, they will not be able to do anything, which is why we call them obligate parasites. Obligate parasites means they absolutely need a host for survival. If a host is not available, they are equal to dead. Okay, so that is obligate parasitism. And these viruses are nucleoprotein. We already know from Stanley's observation that it is mostly made up of protein, right? So apart from protein, they will have nucleic acid. The nucleic acid could either be RNA or it could be DNA. It is present at the center, protecting the RNA or DNA, which is the genetic material. Surrounding that, you will have the protein as a protective outer covering. Now, the protein outer covering is called the capsid. Okay. This capsid sometimes is present in multiple units like this, and each unit is called a capsomere. And the capsomere in different viruses is arranged in different ways. This is tobacco mosaic virus that causes tobacco mosaic disease. This virus has the capsomeres arranged helically. This is a bacteriophage that causes infection, and bacteria, who would have thought that bacteria also will get infections? Bacteria are known to cause infections, right? There are viruses that cause infections in bacteria also, they are called bacteriophages. So here you can see that the protein is somewhat like has a polygonal capsid. It can be helical or it can have like polygonal geometric forms as in this case. So that's about the structure and what the composition of viruses are. I told you that they contain either RNA or DNA. No virus has both RNA and DNA. Either it will have RNA or it will have DNA. If it is a virus that infects plants, it mostly has single-stranded RNA. I said mostly. In most cases, exceptions will be there. If it is a bacteriophage infecting bacteria, then it will usually have double-stranded DNA as its genetic material. If it is an animal virus, it can have single-stranded RNA, double-stranded RNA or double-stranded DNA as the genetic material, which I have mentioned here. Viruses are known to cause a lot of diseases in animals, and even in humans. AIDS is caused by viruses, flu or influenza is caused by viruses, herpes, mumps, smallpox, all of these are viral diseases, right? COVID. In plants also it causes diseases. We saw that in the tobacco plant, but apart from that, mosaic pattern formation on the leaves, some of the other symptoms that you can see in plants which are infected by viruses are leaf rolling like a yoga mat, it gets rolled off, and then the stunted growth. The plants will not attain its usual height, it will be short and the growth will be stunted. Leaves will curl as you can see in this image, and vein clearing. In the region of the veins, chlorophyll will start breaking down and the green color leaf will start appearing yellow at the leaves. That is called vein clearing. These are some of the symptoms that you can see in plants that are infected by viruses. That's about viruses. Okay. Now let's speak of viroids. Viroids are another type of infectious agent. At least in viruses, there was a protein coat and genetic material RNA or DNA. Viroids are even smaller. They are just made up of RNA, and that RNA is also very small and it has a very low molecular weight. Okay. So this was discovered by a person called T.O. Diner while he was studying a disease in potato plants where the tuber, the potato tuber, instead of being spherical or kind of oblong, it became spindle shaped and it was very small. It would have cracks and stuff. So while he was studying that particular disease in disease in potato plant, which was called the potato spindle disease, he discovered the viroids. They are called viroids. Okay. So these are smaller than viruses. They were discovered by T.O. Diner in the year 1971, which is not very, long ago. It is pretty recent. So this caused the potato spindle tuber disease. This is a normal potato, healthy potato. These ones you can see how they are more spindle shaped, right? Then this was found to be free RNA of low molecular weight. They lack the protein coat that viruses have. That's why it is called a viroid. They are just made up of RNA. Okay. Next we have prions. Prions are also infectious agents. They are proteins. These are not normal proteins. These proteins folding, there will be a problem. They are abnormally folded proteins that cause infections usually in the brain. Okay. So these prions are known to cause neurological diseases in humans and other animals, and that is because of abnormally folded proteins. If you compare the size of prions, viroids and viruses and prions are similar in size. Viroids are much smaller. Okay. So, two diseases that prions cause in animals you should know. One is called mad cow disease or bovine spongiform encephalopathy disease. This is found in cattle where the neurological or the system or the brain is affected. These are very abnormal proteins, abnormally folded proteins that will not allow normal brain functioning. They will make more and more proteins abnormal, and the disease will progress like that. Cow or cattle that have spongiform encephalopathy can see how the brain of such organisms, the brain of such infected animals would become porous, like literally spongy. That is why it's called bovine spongiform encephalopathy. So some normal proteins only in the brain that are produced normally, they will only change their shape. Okay, so and then these prion proteins will form and they will disrupt the normal functioning of the brain, as a result of which coordination will become a little difficult and the movement will become difficult and all of that will happen. Similar disease will happen in humans also, that is called Creutzfeldt-Jakob disease. So even in humans, the brain is affected, movement is affected, speech is affected, coordination is affected, depression, anxiety, all of that could also happen. So it's not just like a psychological thing, it is also like literally the brain is affected, okay, because of these prions. That's about prions. And next we have lichens. We are almost at the end of the chapter. Lichens, like I mentioned earlier, are symbiotic associations between algae and fungi. You have algae here and you have fungi. The algae, the partner, algal partner is called phycobiont, it must be here, and the fungal partner is called the mycobiont. Algae, being photosynthetic, will provide food for the fungus. In return, the fungus will provide shelter and help in absorption and pass on the nutrients that it has absorbed onto the, for algae for it to grow and photosynthesize and develop basically. And these lichens are known to be very good pollution indicators. You may have seen these a lot in regions where there is very less air pollution, where the air quality is good, decent. But in cities and towns, you don't see this much because there's a lot of sulfur pollution in the air in cities and towns because of vehicles. Wherever there is a lot of pollution, these will not grow there because these are super sensitive to sulfur related compounds present in the air. So if these are not found growing on trees and rocks around the locality where you live in, be rest assured that the air quality there is not very great. Okay, so that's about lichens. And with that, finally, we've come to the end of the chapter. I hope this was helpful. I will meet you again with the next chapter in the next class. If this helped, let me know in the chat. If you have any doubts, you can ask away in the chat and I will reply to you as soon as I can. All the very best. Thank you. See you soon.